All right, Michael, Bill, are we almost there?
Yep.
Good. And I think we can get going. I mean, it's 8:02. Might as well.
Okay, should I start now?
Yep.
Okay. Welcome everyone to the Carbon Copies Foundation 2020 Summer Workshop event, covering the topic of updating the roadmap to whole brain emulation, part 5, scale separation. What is the necessary level of emulation detail? This workshop event is produced entirely online with the help of our generous patrons and donors, as well as our team of talented volunteers. I'm Bill Nguyen, and I'll be your moderator for today. This event is the 5th installment of our series for updating the 2008 Oxford University Roadmap. For this event, we will be exploring the subject of scale separability in whole brain emulation. For whole brain emulation to be possible, there must be a point of resolution detail of the brain that is less than absolute that can be emulated while satisfying the success criteria for whole brain emulation. Our agenda for today will be as follows. To start off, we will have a short video introduction to scale separability in whole brain emulation. At approximately 8:15 a.m. Pacific time, we will stream our first interview recording. Each interview begins with an introduction to the guest interviewee, so I will avoid lengthy introductions in this listing of our agenda. The first interview is with Professor Steve Potter, Professor Emeritus of Biomedical and Neuroengineering at Georgia Tech and Emory University. Then, at 9:15 a.m. Pacific time, we will begin the interview with Dr. Charles Linson, staff scientist at the Institute for Advanced Simulation at the Helmholtz Research Center for Grand Challenges. Following the second interview, we will begin our live panel discussion at 10:25 a.m. Pacific time, beginning with Dr. Ken Hayworth, senior scientist at the HHMI's Genelia Farm Research Campus and president and co-founder of the Brain Preservation Foundation. At 12 p.m. Pacific time, Dr. Randall Kuna will give the final closing remarks. Our workshop is participatory and is open to all of our international attendees and viewers. You're invited to write questions directly into the chat that is enabled for our YouTube live stream. One of our excellent staff members is monitoring the chat and will alert the panel to interesting and topical questions. More complicated or multi-part questions where it is important to establish the question is properly understood and answered are best asked by directly engaging with the panelists. We make this possible through our call-in number. The number and PIN are shown at the bottom of the screen right now. They are also written in the livestream description and the livestream carboncopies.org webpage. They are 786-540-5631 with PIN 180-618-986 #. If you're calling from outside the US, you may also use the web call-in option at call.carboncopies.org to avoid telephone charges. The process is similar to calling in to a radio show. Your call will be monitored and a respectful exchange is expected. When your question would benefit from direct interaction, I will invite you to call in if you can. If you want to add a note to your question stating that you can call in or that you cannot, that can be very helpful for our moderator. Our many thanks go out to our patrons and donors, to our team members, and to the many experts who make time in their busy schedules to participate in our workshops. We are a 501c3 nonprofit operating with the sole mission of advancing research in whole brain emulation. We appreciate the many contributions that have allowed us to organize events such as these. If you would like to become a monthly donor or to make a one-time donation, you can visit carboncopies.org/donate and donate by PayPal, credit card, bank transfer, or cryptocurrency. Now, before we get started with the first interview, here are a few technical comments that are important for our panelists and as well as others who have joined us directly through our online meeting tool. First, please mute your microphone when you're not speaking. Second, if you have the YouTube live stream open on your computer, please mute the YouTube player to avoid echo effects. Also, unlike previous workshops, we will be presenting our recorded interviews through the online meeting tool. There is no need to switch from the meeting tool to YouTube to watch the interview recordings. Please let us know if any of this is unclear or if you experience any technical difficulties. With that said, we have prepared a short video introducing the topic of scale separability in whole brain emulation. I will present that now. Here it is. Okay.
The most significant question to whether or not whole brain emulation is possible is, can a human brain be emulated successfully without emulating every detail of the organ itself? It might be impossible, and is almost certainly practically unfeasible, to emulate every dynamic detail of every atomic element of a human brain altogether. That isn't even the purpose or goal of whole brain emulation. The purpose is to produce a person's characteristic and personal, cognitive, and conscious experience of being. It's really a focus on the mind, even if that mind is typically the consequence of the biological machinery of the brain. What are the essential aspects of a mind? They are the properties of memory, consciousness, and personality. What is it about the physical components of living brain tissue and their dynamic interactions that make these properties the way they are experienced? The answers to these questions are the success criteria for whole brain emulation. Our world is full of behavior that is described as analog and nonlinear, like the voltage behavior of every transistor in each one of our computers. Certainly, brains are also full of elements with analog nonlinear behavior. Systems like that are called complex and sometimes exhibit chaotic behavior. Behavior that is mathematically describable, but deeply intricate, such as the shape of a fractal. We have to wonder if that means that every minute change at the smallest level might lead to significantly different outcomes at the macro level, where cognitive experience takes place, like a so-called butterfly effect. And would that mean that attempting a separation of scales is even possible to emulate everything above a certain resolution while replacing the hardware below that level? Might there be hope that a system that depends on the reliable interaction of its task-specific parts, a system that needs to reliably steer an animal towards survival, would aim to constrain the range of such effects? After all, even a billion transistors on a chip can reliably run a program and carry out a task when constrained by rules and protocols that impose categories, ones and zeros, and that carry out error correction. We see evidence of such protocols in brains. We see synchronized rhythmic oscillations. We see inhibition of activity following patterns of activity. We see populations of neurons working together in ways that are surprisingly robust in the presence of noise or even damage. Many more examples like that. What then are the protocol constraints that determine the scale of the task for whole brain emulation? Must we reconstruct every neuron, or merely every 10th neuron? Must we reconstruct the geometries of neural dendrites and axons? How much must we scrutinize the behavior of specific synaptic receptors? How should we take into account modulatory effects of diffuse transmitters or electric fields? For which aspects of the embodiment of the brain must we include specific data? Perceiving ourselves in the world is important and provides rich context that our brain is evolved to handle, but the neural control systems that regulate body temperature and heart rate may not contribute more than the relatively simple equations that describe those modulations. It is still very difficult for neuroscientists to trace the path of neural processes that generate a thought. While neuroscience does not yet know enough, we do know some things, and we may be ready to propose a useful version of the principles that are essential and that whole brain emulation must address.
At this time, we present Professor Steve Potter in an interview with Dr. Randall Kuna.
Professor Steve Potter is Professor Emeritus of Biomedical and Neuroengineering at Georgia Tech and Emory University of Medicine. A neurobiologist by training, he's a leading expert on the dynamics of neuronal networks in slice and cell culture. Professor Potter has pioneered dendritic spine measurements, multi-electrode and 2-photon microscopy in neural tissue slice, neural imaging technology, including imaging of GFP-expressing cells, and has built neural probes for neuroprosthesis as well as electro-optical probes. Recent publications include studies of bursts in neural coding, methods of optogenetic feedback control, and hybrid wetware hardware systems, such as the Hybrot, a robot controlled by a computer consisting of both electronic circuits and thousands of rat neurons on a silicon chip. Professor Steve Potter will be speaking on scale separability.
Okay, so thanks, Randall. I very much appreciate you inviting me to talk about these things. These are the subjects that I spend a lot of time thinking about, wondering about, pondering, and which to a large extent helped direct the course of my lab's research since about 1996, I would say. 95 is when I started my postdoc at Caltech. I was there for 8 years building systems for probing neural tissue, and mostly that was in vitro, growing cultures of cortical cells from rats in petri dishes that were instrumented with arrays of electrodes. And we developed the first two-way multi-electrode interfaces with those cultures. So we used them to control robots and to study learning. And the great thing about this kind of embodied cultured network approach that we developed at Caltech and then moved to Georgia Tech to study even further is that you can study learning in an animal, some sort of a hybrid animal that's half living, half technological, and look at its brain while it's behaving. So there are some people who are putting microscopes on the heads of mice and also imaging animals like zebrafish while they're behaving, but that's very difficult, and because of the complexity of the brain, is very difficult, and I would say at this stage impossible to see activity in all of the cells that you're looking, of the circuits you're studying. So we were studying cultures of maybe 10,000 to 50,000 neurons. And in doing that, we were able to actually monitor hundreds and hundreds of them at the same time. And if we did that optically, which we started to do near the end, we were able to see activity in thousands and thousands of cells at the same time, including not just neurons, but also glial cells. So it's interesting to note that there's a lot of information processing going on in glia, and to see calcium signaling in the glial network was very humbling, I must say. So there's that's just kind of a background of the experimental expertise that I have. In terms of my armchair philosophy, I've spent a lot of time thinking about futurism and brain-machine interfacing, reading lots and lots of books about mind uploading and transhumanism and all that sort of thing. I generally am sympathetic to those ideas, and however, I think a lot of people that are writing about them don't know much neurobiology. So one of the points that I tried to make in my 2007 paper about what AI can get from neuroscience is that the computer scientists and others who are working on artificial intelligence would probably be served well by learning more about the actual brain and neuroscientific terms so that they can somehow marry these two things together. Mm-hmm. So I have been reading a lot and studying a lot about artificial intelligence. I was hoping that my lab would get to the point where the kinds of electrophysiology that we were doing and optogenetics would be able to inform the AI researchers how to make better and faster computers, but I wouldn't say we got that far. You know, the kind of computation and learning that we were able to achieve in living networks growing in petri dishes was fairly limited. You know, it was a start, but it wasn't anything that would be better than what you get out of a von Neumann machine these days. I think it's mostly serving as inspiration for being able to study neural networks very much at the networks level rather than the single cell or the whole systems level. You know, it's just another tool in the toolbox out there.
Yeah, so you've been using biological, in vitro, cell culture neurons and networks as computational devices rather than, say, for example, simulating or emulating, making models of networks of neurons that you might find in.
Yeah, I think it's really interesting that we did both. We one of my graduate students, Zanis Chow, made a 100 neuron integrate and fire model, which was fairly simple. It had about, had the right proportions of inhibitory neurons and excitatory neurons, and it had maybe about 1,000 connections per neuron to the other neurons nearby. And there wasn't much to the model, there wasn't much biological complexity to it. And yet, despite that, we saw amazing similarity in the kinds of bursting dynamics in these model networks that we saw in our living culture dishes. So much so that they allowed us to design our experiments that we carried out on the biological networks. So we first tried a certain kind of learning algorithm, you could call it an evolutionary approach to training these networks in our model networks in simulation. And when it worked in the simulation, we said, okay, let's try it out in the living networks, and it worked, and it is a testament to one of the points that I think you guys will be happy to hear, is that you don't need to know everything to do some kind of brain emulation. You can pick and choose what level of detail you want to include in your simulations to get some sort of behavior that you're interested in emulating. And for that reason, I think it's a very good idea to have your goals well defined at the outset. I think when I hear a lot of these folks talking about brain emulation, I think about the folks that made Deep Think and Hitchhiker's Guide to the Galaxy. Did you read that one?
Of course, yes.
So they it's a classic, and they failed to define their question clearly. They said, okay, give us the answer to the question of life, the universe, and everything, and it came up with 42. And if you fail to define your question carefully, that's what you end up with, a fairly worthless answer, and then you have to go and build another bigger, better computer to try to figure out what the actual question was in the first place.
Yeah, that's actually the principal problem, I think, with a lot of the conversation that's going on right now, where whole brain emulation or brain emulation or any of those things are concerned. And in fact, the reason we're doing this bit right now, this part that's going to be a part of our next workshop on scale separation and on the resolution at which you want to do an emulation, is because we're still working on clearly defining that part of the updated roadmap that is supposed to be all about success criteria for whole brain emulation,
right?
Which is not exactly the same thing as success criteria for mind uploading, but it's closely related, because if you want to have successful mind uploading, then you want to do whole brain emulation in a specific direction, which is a different direction than, say, for example, the type of emulation that you would do if you're trying to explore the causes of Parkinson's disease or something like that. And I think it's important to have an initial chapter, basically, that just talks about what are the success criteria. Even if you cannot pin them down perfectly, if you can't say, these are the criteria because, then at least you can either talk about a consensus where most people would agree, or you could talk about some minimum requirements, and so forth. So that's really where this comes in. Now, I'm kind of curious, because you mentioned that it's great to see that you can emulate even when you can emulate to some degree with some goal in mind, even if you don't know everything. You also mentioned glial cells before, and I'm wondering, of the things that you have encountered, before we even dive into all of this, what would be, say, something that you think was particularly surprising, something that you didn't expect?
Yeah, well, the thing that blew me away the very first time I watched a time-lapse movie of neurons growing, so I've made a lot of time lapses of neurons growing, and when you watch a neuron under the microscope with your bare eyes, what you see is something that's moving about as fast as the minute hand on a clock, so it's not, you don't see a lot of obvious activity there. And then if you take a time-lapse movie over a matter of hours or even days, in some cases we did weeks, you see a lot of activity. And what blew me away, what was so surprising, was how each neuron in the dish seemed to have its own personality and its own little agenda. And I felt as if I was looking down on a party from above, and they were reaching out and making connections with each other and pulling back their connections in some cases. And I sort of lost my individuality at that moment. I suddenly realized that I was a collective of many, many cells interacting, who are just somehow operating these vocal cords, you know, and producing speech that sounds like it's coming from one entity of some sort. But really, it's a group of things that have this amazing ability to get their activities coordinated. So I suppose that was the first most surprising thing that I came across doing these types of studies, was just the sheer complexity of it. You know, I think unless you have spent hours looking through a microscope at actual living cells, like I have, you just really can't quite appreciate how complex brain tissue is. You know, and even this was fairly simple tissue that was growing in a monolayer in a dish. When you start to look at specimens that are more intact, or actual intact brains under the microscope, just mind-blowing how complex it is. And then you can start to look at when you have these things on an array of electrodes, or you've got them hooked up with voltage sensitive dyes, the amount of diverse dynamics that you see is just doubly mind-blowing. So I think that's the kind of thing that computer scientists don't really appreciate. You know, when they say, okay, we're going to emulate the brain by just pulling out the layers that are important here and modeling them, they don't really realize how immensely complicated this thing is between your ears. Even in the tiniest of brains, you know, the little fly brain that can steer a fly and allow it to mate and avoid predators and find food and all the things that a fly does. We just have nowhere near the ability to understand, and certainly not to model that level of complexity.
Yeah, and I mean, oftentimes when you see images of you know, what you might see under a microscope, then it's something that's fluorescent or stained, and there's only 10% of the neurons that are actually stained, so the entire area looks kind of empty with just a few cells in there.
Right.
But that's not the reality either.
When you see those serial EM sections of the retina or something that just every little nook and cranny is filled with another neuron doing its own thing.
Yeah, and I was also, it's interesting that you mentioned as a surprise, seeing what was happening when you watched the cells grow under the microscope, because I would say that was probably also something that I found surprising back in, say, 2004, when I was working with Van Pelt and Raumakers and others there who'd like to, you know, work on trying to model the growth of cells. And it was amazing to me to see just how much of the blueprint of how the brain evolves or how the brain grows is just something that the cells do without any specific input whatsoever, that they just connect in a particular way, and then you'll start to see typical rhythms emerge from spontaneous activity that haven't been put in there through any learning or anything. And that's something that when you think about artificial neural networks, that maybe there isn't a lot of attention being paid to the importance of the blueprint of the human brain, and that blueprint makes us already more capable in some ways of understanding things that are in the world that we evolved into. So
obviously the genes that the neurons are carrying and glial cells, we must try to continually not forgetting the glial cells. The genes they're carrying are telling them something about what to do, but it's also very much based on the environment. You know, we actually were able to observe cells switching from being excitatory to inhibitory early in their life, based on the kinds of inputs that we delivered to them. So it's not all cast in stone, at least at the late term embryo stage that we got our cells from. And what that tells me is that when we finally do understand even what this word blueprint means, it's going to be more complicated than just, oh, we can read the gene sequence and we now know how this network is supposed to hook up and how these particular cells are supposed to behave by expressing these genes. it's going to be a complex interaction between those genes and the local environment the cells find themselves in. And because neurons are unusual, they also get influenced a lot by non-local environments. They have axons that can reach, in some cases, many meters across a long animal. And they can be influenced by signals traveling those great distances.
Indeed. So tell me something about glia.
So, well, again, with a recurring theme here is that there are many different types of glia. You can see if you have a stain that is selectively taken up by glial cells, I think flow 5 or something like that, one of these flow dyes was selectively absorbed in glial cells. You can see a lot of different morphologies, and in some cases, you have these very, very branchy cells that you might think they're astrocytes, which is one kind of glial cell. And then you do something that stresses the culture. Maybe you forget to feed it for a couple of days, and those things transform into reactive astrocytes. And they pull in most of their processes, and they become very motile, and they roam around, becoming the garbage collectors of the network. And so in some cases, we saw these networks go from a fairly comfortable, stable state with the glial cells all interspersed in between the neurons, and then they just sort of mobilized into action and started to do something completely different. So it's a lesson for those who like to do EM, and fix tissue and slice it up and look at it in detail under the microscope. You're only getting one tiny little snapshot of what that tissue was like at that moment that you threw in the formaldehyde. Those things are dynamic and changing all the time, and they might be changing rather dramatically into something completely different, and you might not have caught it in the act of changing. Scott Fraser used to say, he was one of my postdoc mentors, used to say that it's a lot like, trying to learn the rules of a football game from just looking at a few photographs of a football game. You'd be much better. It would be pretty confusing, and you'd be better off taking a video. You might have some hope of actually learning the rules by watching a video of a few games rather than just some snapshots from football games.
Okay, so that still sounds like glia are doing these somewhat non-specific things, or am I interpreting that wrong? As in, it sounds like they're still doing a mostly support function.
Yeah, so those reactive microglia
that.
probably are, but the ones when they're in that sessile branchy state, I suspect they're doing localized computation. And the reason why I believe that is because we've made some of these calcium imaging movies of signals going through glial networks after the neurons either had died off or there were very few neurons in the dish. The glia keep on going. They're much heartier than neurons because they can divide. And what we saw was just very diverse, complex dynamics in the calcium signals that these cells were producing spontaneously. We weren't stimulating them or anything. And the trouble is that electrodes don't pick up these signals. So neuroscientists, by and large, thought that glia were uninteresting because when they poked an electrode into a brain, they hear the neurons, and the glia are signaling. They're doing lots of signaling, and they just don't make action potentials. And that kind of signaling that they're doing is just mostly chemical. And we need to have much better sensors than we have now for picking up chemical signaling to be able to really understand and decipher what the glia are doing. So I think...
So to just poke at that a little bit.
Okay.
Okay, let's say that we have a neural network or neural circuit. The neurons are communicating with one another through mostly the spiking, and then you've got your glial cells that are producing chemical signals. And let's say the chemical signal is changing dynamically. So, someone who's poking an electrode in there might say something like, well, I don't need to care explicitly about the glial cells because I'm picking up the effect of their chemical signaling as it impacts the membrane potentials and eventually the spiking in the network. So if I don't model them explicitly, I'm not losing something. Is that wrong? Is there also a separate signaling pathway through just the chemical signals that never becomes part of the neural network? Is it not the neural network that ultimately is really governing the output of the system?
Yeah, I think that's the key question to ask, Randall, that are there glia that are effectors, And I think so far anyway, the answer seems to be no, that the things that cause muscles to move and glands to secrete are neurons. The glia are there at some earlier stage in the sequence of events. So I think you're right that you might be able to ignore them safely if what you care about is the processing of the system. Okay, now you may want to be, go back again to trying to define your question more clearly. What exactly are you trying to emulate? If you're trying to emulate plasticity and how learning happens, which is what we were very interested in studying, I think you do need to study the glia. So for example, we saw some really interesting dynamics in the changes in the propagation speed of signals, of action potentials in our culture dishes. And I don't think that these neurons were myelinated. I suspect, we were taking cortical cells at a stage where there certainly wasn't myelin already. It hadn't formed yet in these animals. But it's possible that there certainly were oligodendrocytes and other glial cells that would form myelin in our culture dishes, but we didn't observe, for example, tracts of white matter forming. That said, I think the glia are there somehow modulating the propagation speed of action potentials along various unmyelinated axons. How they do that is something that needs to be studied, but we had ways of studying propagation very precisely down to the tens of microseconds time scale by basically stimulating on one part of an axon and recording on another part without there's no synapses involved here. These are just what we would call direct action potential recordings. And these values would change with plasticity, with the kinds of plasticity that people think are all synaptic, that are learning related, that are necessary for learning to happen. The propagation speeds would be changing. So, maybe it's the glia, maybe it's not. Maybe it has nothing to do with the glia. It could just be the distribution of ion channels in the membranes of the neurons. It could be how the time course of opening and closing of ion channels. There are a lot of other possible explanations, but I suspect that the glia are involved in fine-tuning these delays, because I think that they are something that the brain uses to do a lot of its calculation, fine-tuning its delays.
How much did the propagation change? What range of differences did you see?
Yeah, well, in the case of these are not very large distances we're talking about. They're propagating only on the order of maybe 3 millimeters or so. And we saw 10% changes. That was probably the biggest change that we saw, that the speeds would change on the order of 10%. And in both directions, slowing down and speeding up. And these would they were causing changes that neurons would be sensitive to. So the time scale at which neurons seem to care about, at least in our culture dishes, was on the, say, 1 to 10 millisecond time scale. In other words, we found that networks, when we observe some kind of plasticity, when we were able to induce a change in the network's function by producing some kind of pattern stimulation, and then observing how the network responds to it and noticing that it changes, the kinds of stimuli that the network could be changed by were those that had structure on the scale of 10 milliseconds or so. And so the changes that we are observing in the network's own propagation speeds would have been noticed by network effects.
10 milliseconds is definitely significant. I mean, it's the kind of thing where if you have two different regions that are communicating with one another, it can be the difference between an offset where they manage to do that or not. So
yeah,
that's quite something. If you don't mind, I'm going to squish together two of the questions that I had prepared because I think that they may go together well right now. well, actually, really one of them you just mentioned, which is to try to define better what the goal is of emulation. So I'll try to define it at least in the very in a sort of broad way that that I've mentioned it in introductions to this workshop that's coming up, and that is that the emulation that we're aiming for is an emulation that preserves what we might call the cognitive experience. So it's not about studying disease. It's not about knowing which synapse in your brain has just activated, because we typically don't experience that as part of our cognitive experience, but cognitive experience in the sense of being able to process what is going on around us and still having goals and retrieving memories and consciousness, those things, that I would say is the goal environment, I guess, that we want to be in when we're talking about brain emulation of that kind. And sort of looping in one of the questions that I sent you, since we're talking now about milliseconds differences in propagation speeds and how the glial cells can affect that and so forth, that borders on the territory where select people, I'll just say, will say, for instance, well, we can't.
Yeah, so I think I'm delighted that you're asking these questions about what level you want to emulate and trying to refine your goals there, because I think that's the key to making real progress on this. The more well-defined you are in terms of what you're trying to accomplish, the more you will be able to winnow away some details that may or may not matter, and I think we proved that that's possible with our 1,000 neuron simulation, that we were interested in bursting dynamics and whether or not bursts could induce long-lasting plasticity, and if so, how do you express that and how do you measure it? And our model network worked with very few details, very few biological details. So I'm very hopeful that we don't have to be defeatist and say that because of the rich dynamics that brain circuits produce, we'll never learn anything and we'll never be able to emulate anything. I think the key is just keep trying to work your way up from something fairly simple. Now, there's what I would call the Eve Martyr effect. She's somebody who has studied lobster circuits for her career and the stomatogastric ganglion, I believe. And she has convinced me that it's kind of futile. The way she said it is you can know every connection in the circuit. They basically studied this so well. They know where every synapse is in the circuit, and they still can't figure out why it behaves the way it does. And they concluded that it was all down to neuromodulators, that when you sprinkle on different types of neuromodulators on these circuits, they have completely different dynamics. They behave in a different regime of their dynamic landscape. And so I think that the way forward, the thing that I see, there are people obviously who are working on this, but not enough of them. The way forward is people need to develop new tools for studying those complex dynamics, like basically building a microscope that sees neural dynamics in a network level. It's one thing to stick an electrode in one cell and say, aha, I see it oscillating, but the kinds of dynamics, even in a lobster stomatogastric ganglion, were immense and very rich and amazing. Even in C. elegans, we've had the circuit diagram for C. elegans since the 80s, I believe. They sliced it under the EM, and they've measured where every single synapse is in this 300 neuron animal. It's a millimeter-sized worm. And they still don't understand how it learns, behaves, and responds, and does all the amazing things that this worm does. So you could be defeatist and say, I'm going to give up. It's hopeless. But I don't think that's what we should do. I think what we should do instead is develop more tools for looking at networks. For example, voltage imaging. If you could look at C. elegans or a lobster, or preferably an animal or human that's a little more complicated brain, with voltage sensitive imaging that was good down to the millisecond timescale resolution, I think you could start to build a dynamics microscope that would be able to classify different types of dynamics, and how do circuits switch from one type to the other, how do they make discriminations, how do they make generalizations, how do they make different types of bifurcations in their behavior. You know, there's lots of dynamical systems that people have been studying that are physical systems, and there's a lot of physicists out there that have developed tools for studying them. I think those guys need to up their game and say, okay, now we're ready to attack the brain and apply these tools to neural circuits.
That's a really interesting direction that you just took there, because I think it's very important to understand that there's a difference between a complex system that simply is, like say the weather, and a complex system that is attempting to achieve a specific thing, like the brain, which is trying to keep us alive.
Yeah.
And that's, you know, I mean, for example, this is where I think that sometimes looking at C. elegans or looking at the lobster ganglia, that can be deceiving, because you're looking at the nuances of that little analog circuit that is not much like our brain, in the sense that it doesn't have these gigantic areas with populations of neurons trying to communicate with one another.
Right.
You just mentioned bursts and bursting and its effect on learning. Not just on learning, of course, bursts are also very important for communication between areas in the brain. I think that's, so if I'm going to go out on a limb, I'll say that is something the brain does and has to do. It has to have certain principles, protocols of communication that provide for it something that is more reliable, more repeatable than, say, the individual spike at a synapse or at a neuron. And that is, I think, one of the things that really has to be cataloged, be developed in the way that, you know, we took certain principles and moved them into artificial neural networks. You know, we said, okay, populations of neurons that have attractor properties or something. That's a great thing to have. But there is still a lot in there that hasn't really been sort of written up as, okay, these are the principles of how the brain makes itself predictable enough so that this region can talk to that region.
Yeah, so population coding, I would say, is a field that is we're still in its infancy. You know, if you think about Galileo looking at other planets, looking at Jupiter, say, that's about where we're at right now. We're looking through a refracting telescope at Jupiter. That you know, we need to be able to send Pioneer or Voyager out there past Jupiter and take close-up photographs to really appreciate just how amazing the planet is. And we'll see all sorts of new things when we develop those new tools, like a spacecraft. You know, so what would be a spacecraft for dynamics in neural circuits? That's a good question. I think that we need to, one of the biggest stumbling blocks here is the terminology and the language. You know, that the neuroscientists are not used to thinking in terms of these dynamics. The physicists are not used to thinking of neural circuits and how complex they are. And we just need to do a little bit of cross-pollination, which you guys are doing. You know, getting people talking in both sides there. I wanted to mention something about population coding. You know, think about a bat catching a moth as it's flying with echolocation. These animals and lots of animals can do sound localization amazingly well in the dark, and they're measuring interaural time differences, the time that sounds take to come to their ears, on the scale of a few microseconds. And there's no neurons that can fire at those frequencies. So they're obviously using populations of neurons to do this very amazing discrimination job. So yeah, there are a few circuits where population coding has been at least proven, if not understood. You know, so certainly we can point to quite a few circuits. Like, you know, I think Georgopoulos and the monkey motor cortex was the first one that I heard of, where that you record from an individual cell. It doesn't tell you much, but when you have 100 cells, you go, oh, wow, I can tell exactly where that monkey is going to point because it's population code. So just develop tools to be able to focus in on a circuit well enough and record as many neurons as you need to say, here's what the population code is and here's what it's encoding. I think that's an example of the kinds of things that I would say are like a Pioneer or a Voyager spacecraft to fly by Jupiter there.
Mm-hmm. Yeah, there's also, in addition to those tools for recording and observing, which you want to be able to do that in detail, but you also want to be able to see entire populations. In addition to that, there's also this problem of modeling, where most computational neuroscientists work with what are clearly toy networks, in the sense that either they're designed entirely by hand to do a specific thing, or they are trained to try to work like something they found in the data. It might even just be a GLM or some other very standard learning trainable model. And in those cases, they're usually constrained to working with a very small number of data, or let's say number of dimensions that they can use, because the problems tend to explode very quickly when you're trying to. Yeah, you're smiling. I was just going to ask you about your experience there and how you try to control for that. How do you try to keep that manageable?
Well, I'm not any good at modeling. Our simulations that we did in our lab were able to be run on a single laptop computer. So we weren't pushing the bounds of what we could do with modeling. We had servers in the NeuroLab at Georgia Tech that I always imagined we would be using for that purpose, but honestly, we never got that far. We didn't make modeling a priority in our lab, or simulations, I should say. There are others who are really pushing the bounds, and I think for these things to blow up is a very common thing. And at that point, you just have to say, okay, what can we do without in our model here? And all you can do is just really focus your question and say, I really only want to study this one thing. I just want to study how do propagation delays vary when glial cells get thrown in the network. Or I just want to study how do bursts work together to make information transfer over large distances more reliable. If you have your questions very precisely defined like that, I think your model, you can make a model that doesn't blow up. You can put as much detail that you need specifically just to answer that question. If you want to say, I want to emulate a brain, and I want it to have some sort of high level conscious experience that's unique to humans, we're in trouble there because we just know so little about where that comes from. I think the cognitive scientists still have the best approaches with their, completely non-biological approaches to understanding these things. Just to say, okay, let's make a blackboard model. That's as good as anything that neurobiologists are coming up with in terms of studying the dynamics that lead to the feeling of me and where self comes from. So, some people would take issue with me on that, but I think I don't want to give up on those things. I think they're really interesting questions, but I'm most inspired by Douglas Hofstadter, who I think really has done a fantastic job of saying, okay, creativity, there's a really hard problem. It's something humans are really good at. Where does it come from? Let's try to make a model of a creative system. And he did it. His grad students and him made these amazing models where they did, for example, taking a font style and showing just a few letters in that style and saying, now make me the whole alphabet in that style. And the computer program could do that. And he learned a lot about what is creativity and analogy making from doing that. So without using a whole lot of biology, there's not a whole lot of neurobiology involved in that process. And eventually, I have confidence they'll come together. They will meet
perhaps the only thing we need to do to discover how consciousness really emerges is to play around and make an artificial consciousness, come up with something.
So I think we've already done that. I think one of my pet peeves is people who are very binary about consciousness, and think that it either have it or you don't have it, or it turns on when you wake up and it turns off when you go to sleep, or that humans have it and cows don't, or that cows have it and snakes don't. That I believe there's a whole spectrum of consciousness, and if you believe that, like I do, there is very simple systems that are conscious that we've already made on computers. You could argue that your computer is conscious. You know, it's conscious of me tapping away on the keys. It's conscious of me talking into its microphone or sitting in front of its camera. It's conscious of a lot of things going on the internet. Whenever Microsoft decides there's a software update, it seems to get really anxious about it and want to remind me constantly about it. So that kind of consciousness is not what most people are thinking of when they say, you know, oh, where does consciousness come from? But I would argue that it is. It's that human consciousness is just a huge pile of little hacks that all do various little jobs, you know, and when you pile them on top of each other and when you pile them on in great numbers, you end up with something that's amazing, like what we have in us. I think a lot of it was all just as a learning mechanism. You know, there's plenty of animals out there that don't seem to be terribly conscious, certainly not of themselves, not self-conscious. I'm looking out on the cows and fields out there right now, and you know, they're walking around naked. They mate in front of each other. They pee and they poop in front of each other. They have obviously no sense of embarrassment the way humans have. Where did we get embarrassment from, and why do we have this, and what purpose did it serve? You know, that's a big part of our self-conscious experience is embarrassment. It obviously serves some evolutionary purposes, and if we focus on those and we want to study consciousness, I think to divide it up and to break it down into little tasks like that, saying, you know, what does it serve, then we'll understand a lot better. That's what Christoph Koch really did. He said, well, let's just look at visual awareness, you know, stop talking about consciousness. Let's just say consciousness equals visual awareness. If you don't agree with me, you know, go away. You obviously don't care about what we're studying here, and I'll talk to the rest of you who do care. And he was able to make great progress by doing that, you know, and saying we're studying the visual system. Where does it get these ideas for what things are out there in the world through its retinas?
Yep. Yeah. So I guess since we're talking about consciousness already, this means I can again combine two questions I had. One of them was really to try to figure out if you felt that consciousness was any different than, say, some other part of our cognitive experience, like, say, visual processing or something of that nature, in the sense that it would require something different from the underlying neural mechanism, from the neural correlates, let's just say. Would it require something else? So that's part of the question. And since we're going there anyway, my overarching question is, if you had to place your bets, how deep would you have to go? What needs to be modeled? What resolution are we talking about to capture cognitive functions and experiences?
Yeah. So with all the caveats that I said already about, you know, looking at consciousness as a spectrum and studying it in a very defined way, the kind that people seem to care the most about, this kind of feeling of how it feels to be me and that sort of thing. I think that, first of all, I'm a materialist. I think everything that the brain does is thanks to the physics of brain cells. You know, there's no magical substance in there. I even go so far as to say I highly doubt that there's any quantum effects that are important. You know, as I say, the jury is still out on that, but there may be something that's subtle that we're totally overlooking, like ephaptic signals. You know, this is how do electric fields affect the dynamics of neural circuits? Because neural circuits make their own electric fields. You know, you can obviously record electric fields through the scalp and the skin in the skull. And so the neural circuits are producing electric fields. And we also know from, you know, putting electrodes on people's heads and doing TDCS that you can influence neural circuits with electric fields. So it makes sense that they probably evolved capacity to influence themselves with their own electric fields. This is something that very few people have studied at all. You know, other than just to say, yeah, I think there is ephaptic signaling that doesn't happen through synapses, it doesn't happen through gap junctions, it doesn't happen through hormonal modulators, it's too fast, it's definitely electric field signals. So there might be some layers like that that we're just missing in our thinking about consciousness and what makes brains do what they do. That said, I suspect that if if we wanted to make a model that was conscious, we might not need to include that stuff, that we probably have the tools and the understanding right now to model something that is conscious to some level and has some sense of self and some sense of what it feels like to be me. The big trouble is, how do we know it does? It's a first person perspective problem that's slowly being solved with fMRI, but fMRI has a very big chunky spatial resolution. Its time resolution is even worse. So if we develop some kind of imaging system that was down to the single cell level, or preferably even smaller, that could look at activity in every cell in a brain or a whole neural circuit, I think we could start to get at these questions much, much better than what we're doing right now with fMRI and in vivo recordings from animals with lots of electrodes poked in their brains.
Yeah, so I guess another way of looking at or taking another angle on this question of what resolution is to ask, is there anything besides that which affects processing in the neural circuit that would be really essential for cognitive function? And the reason I'm asking it this way now is because, we talked about chemical signaling by glia.
Yeah.
And we said, well, they probably only have an effect in the sense that they affect the neurons, which are calculating stuff and producing effects. How about these ephaptic couplings? We could say they also only matter in the sense that, well, the neurons are generating them and other neurons are picking them up. So again, it's the neural circuit that is talking to itself.
Yeah.
In this case, right? It's not really something entirely different that we have to include there. It's just part of that the signature of how are the responses being affected by the other responses.
Yeah, you can make great headway just by looking at the input-output function of a system. Call it a black box like Ted Berger has done with, small chunk of the hippocampus. You can probe what its input and outputs are without really understanding a whole lot about what's going on in between there, and emulate its function that way. I think the thing that's missing, you asked, is there something else that we need to include? And I think that is the environment. I think that you stick a neural circuit in isolation, sensory deprivation, and it ceases to function very quickly. We observed this with our culture dishes. And when we embodied them, when we gave them a sensory system, an artificial sensory system, and the ability to express their activity as behavior in a closed loop fashion, their dynamics changed dramatically. So I think if you really want to study a neural system, you have to embody it, and you have to, give it an environment in which to behave, preferably one that it evolved to behave in. I think who are those folks? There's some folks that Bill Bialik maybe who study blowflies and information processing in their visual circuits, and the bit rate that they were measuring in their visual system was immensely much greater when they went out to the forest and measured them in the forest where the flies live than it was in the lab when they just showed them, they waved their hands in front of a fly. Here's a fly glued to a pedestal in the lab. It's not able to move. It's not flying around. And then they went out in the forest and they built this amazing fly spinning device that could spin the flies with angular speeds similar to a fly flying. And suddenly their visual system was encoding much more information than it was otherwise. So I think this really brought home to me the point that the environment matters, and the kinds of sensory inputs that you get in a real environment that you evolve to process are what really matter if you want to understand how a system works. So if you want to understand consciousness and the feeling of what happens to be me, what does that mean? Your simulation has to be in a virtual reality that's kind of real.
Don't study it while you're under anesthesia.
Yeah.
No, this is a good point you're making. In fact, whenever we talk about Ted Berger's hippocampal model, it's really the same caveat that comes up. You want to build a model of that piece of circuitry from CA3 to CA1, and you certainly know a lot about the underlying blueprint of typical circuits there, and we've got some recordings, maybe 60 to 100 on one end and 60 to 100 on the other. which is of course extremely tiny. And then the circumstances in which the model is meant to work is extremely constrained. It's an experiment where the patients are seeing some images and then they have to remember which location of the screen they saw them on or which image they happen to have seen before. That is not a natural environment. That is not your daily context. And so the model hasn't seen, learned, or tried to work with the types of signals that you would encounter in those circumstances. Yeah, that's that richness that's really missing right now.
Yeah. So if I were to make one argument for all of the modelers out there is really think about embodiment and the environment in which the model is living.
Mm-hmm. Yeah, I think that's actually something that maybe you can say more about that when you say embodiment, because people often think embodiment just means, oh, so somehow it's important if we stick this in a robot or something like that. But maybe they're not really thinking through what embodiment means, because just now we were talking about embodiment more in terms of signals and the richness of the signals that you come across, right?
Yeah, well, there's a lot of computation that's not done by neural circuitry, but by the physics of the embodiment. You know, the dynamics of running and flying in flies and stuff like that are computed. You could argue they're computed by the masses of the wings and these halteres that are little weights that are swinging next to the wings. These things have resonance frequencies that allow them to fly and to react to certain muscular inputs in certain ways that allow them to be aerobatic. You know, so there's a lot of nature will take computation wherever it can find it, and I think we have this kind of neurocentric point of view that neurons are the things that are doing all the computation, but I don't think that's the case. I think embodiment is really important. And, you know, I think a lot of these kind of transhumanist thought experiments involved, you know, slowly replacing one part of the brain with an artificial thing and saying, you know, look, you wouldn't notice this change and you wouldn't know this change. But I would argue that, yes, you would notice every little change. You know, if I have a Band-Aid on my finger for a day, my somatosensory cortex is probably changing, because at the end of the day, when I take it off, it feels weird. There's something weird going on there. You know, so the embodiment is constantly interacting with the neural circuitry to optimize whatever computation it needs to do, and we need to take that into account.
Indeed.
Yeah.
So is there anything else before we finish up that you think that we didn't touch upon that we should have? Something that you think is important?
Let's see, I suppose that the thing that, you know, so I packed up my lab in 2015 for a variety of reasons, but it wasn't because our research wasn't going well. We were doing very well, and we actually had a grant that was only halfway finished. We basically finished the goals of the grant in the first year, and those were to make a closed loop optogenetic system for interacting with culture dishes for studying homeostatic plasticity. And this was the work of Ming-Fei Feng and John Newman. And they did some fantastic studies of how networks change their synaptic weights if they're not getting enough input, or if they're getting too much input, they're constantly dialing up and down their sensitivity. And this is a model system for studying all sorts of homeostatic mechanisms, like the reasons why we develop epileptic seizures, the reasons why we have tinnitus or chronic pain. And homeostasis is also a good thing in a lot of cases. You know, it helps you in many ways. So anyway, the thing that we haven't really touched on is how important and exciting optogenetics is. That nowadays we can stick genes into neurons that allow them to be light sensitive. And the reason why that's really cool is because, number one, we can use different wavelengths of light to do different things. We can stimulate neurons with one wavelength and inhibit them with another. We can label certain subpopulations of neurons with one construct that's sensitive to one color of light. So we can just excite you know, the basket cells and the chandelier cells with one wavelength, and we can excite the pyramidal neurons with another wavelength. So it's compared to stimulating with an electrode, it's just amazingly more exquisite and detailed input on the input side. So that's on the input side. We need to have similar advances on the output side. Right now, there are genetically encoded voltage indicators for neurons, but they're not very good. They require a lot of light, which tends to kill the cells. So you get only a few seconds of good recordings before you've blasted the crap out of the neurons. And or the signals are too, you know, have such a poor signal to noise ratio that you can't really see individual action potentials. You can only really look at bursts or other very large signals. So you kind of got to take your pick, or you can go with calcium signals, which have a much better signal to noise ratio, because guess what? Calcium signals are about 10 times slower than neural action potential signals. So you can integrate the photons over a longer period of time, and you can get a much better signal, but you're missing out on a lot of the details of the computation. So anyway, my soapbox here is that we're going to get much better tools for studying the brain when we really develop these optogenetic tools in both directions, on the input side and the output side. There's another really good reason to do this, which is that electrodes are not very biocompatible. You know, if you stick an electrode into a brain, it's very quickly engulfed by glial cells that are trying to dissolve it. And in most cases, they succeed in ruining the electrodes. And if they don't, they at least succeed in muffling the sounds that you can pick up from the electrodes and your ability to stimulate as well. Whereas optical fibers are pretty biocompatible. You know, glass is an extremely inert substance, and brain tissue does not seem to mind having optical fiber optics implanted in it. And even if it does get encapsulated by glia are quite transparent, and the light shines right through them. So I think the long-term goal for neural interfaces and in most studies of the brain is with optics, and especially if we want to look at large-scale dynamics of entire networks where we see the activity of every neuron in the network.
Yeah, that's a very good extra comment there. So that almost felt like you might have some commentary on the tip of your tongue about the recent Neuralink demo and where they're heading with things. But you don't have to go there. It's okay, unless you want to.
Electrodes aren't going to go away tomorrow. And in fact, our lab used them along with the optogenetics very effectively. You know, we made a sort of hybrid system that was both electrical and optical. And so they're not going to go away. They'll still be very useful for a long time to come, and maybe forever. However, you know, our understanding of how to make genetic constructs, and especially how to get them into mammalian neurons and humans especially, is limited. You know, people aren't willing to take the kind of viral constructs that we used in our culture dishes to put these genes in there. Gene therapy is moving along, and there are many other incentives for making gene therapy happen. So I have confidence that eventually that thing, you know, everything will be worked out, and all of us will have some kind of genetic constructs in our brain cells that make them light sensitive, at which point we're going to have to all wear foil hats to make sure that the sunlight does not affect us too much.
Maybe, but that would be so wonderful, though, for all of us who want to connect with the brain. Yeah. But in the meantime, I think what Neuralink is doing is probably a very good idea. And in fact, they're upping the number of electrodes by at least a factor of 10, if not 100. To compare to what's been done in big animals. So I'm very impressed by them. Indeed. So thank you. I think I've run out of any questions for the moment, even though clearly the topic isn't nearly done, because we still have to, you know, work our way towards a better definition of exactly what the goal is of brain emulation, and we still have to work our way towards examining more carefully statements about, say, does everything that we care about happen within the neural circuit, you know, embodiment notwithstanding, and can all these other effects that we see there, all these other modulators, can they just kind of be wrapped into that by looking at it as a black box? All these questions still need more detailing, so we're definitely not done. But
Yeah, well,
I am done with my questions for today.
I'm looking forward to the panel discussion in which we'll get live questions from the listeners. Correct? Yeah. So they'll help us, hopefully help us refine our questions. You know, the kinds of questions that they're interested in can help researchers decide what to focus on next.
Indeed.
Many thanks to Professor Steve Potter for that great interview. Next up, our second expert, Dr. Charles Linson, in an interview with Anita Fowler and Michael Ulrich.
Dr. Charles Linson is a staff scientist at the Institute for Advanced Simulation at the Helmholtz Research Center for Grand Challenges in the town of Jülich, right where Germany, the Netherlands, and Belgium meet. Dr. Linson's expertise is in computational and systems neuroscience, with a specific focus on large-scale simulation and neuromorphic systems. In that domain, Dr. Linson is a co-developer of the NEST simulator and the NestML domain-specific language and toolchain that aims to make the specification of dynamical neuronal models simple but expressive. Dr. Linson also curates a gallery of astonishing neuroscience images by artists and photographers worldwide. Dr. Charles Linson will be speaking on scale separability.
Yeah. So yeah, maybe as a segue then, I guess we're looking for some theories to give us some explanations. So to come to this question of level of detail, you know, I guess the main question is, you know, what is the level at which we want to understand the brain?
Mm-hmm.
And yeah, we've had obviously much progress in deep learning and a lot of tricks associated with that, like, you know, the dropouts and things like that. A lot of interesting stuff, reinforcement learning in recent time. And I think maybe many of us are hoping that will be the level at which we can decompose the brain, that we have some functional modules, maybe many functional modules, that we can somehow wire up. I mean, I've seen some really magnificent graphical user interfaces for designing deep nets and connectivity patterns between them, and then to yeah, in different learning. Yeah, you know, the whole parameter space of deep learning. Yeah, you know, I guess maybe this is a kind of idealized view that then we could kind of use, you know, this kind of designed approach, maybe, you know, to some degree, or maybe largely data-driven, data-constrained. You know, we have to relate to the actual real brain, which is giving us a very good working example of you know, what it is we're trying to do, except in biological matter. But yeah, so I mean, this level of abstraction, we have some modules that we connect together according to maybe then, you know, some tractography data. I'm thinking of these original hierarchical views of the cortex, for example, where you have these beautiful, you know, boxes and a sort of a cobweb of connections between them, but it's a structured cobweb, and we can place cortical modules in a hierarchy, and it all seems perfectly decomposable at this level of abstraction. And personally, I really question that. So for me, kind of my personal view right now is that we're gonna have to look at some detail at the biophysics. So the brain has what 10 to the 11th neurons. So we're at least gonna need 10 to the 11th state variables, dimensions in our dynamical system. And then we have the synapses. We have 10 to the 14th or 10 to the 15th synapses. So that's a real bottleneck. And especially accounting for dynamical synapses, where learning is continually occurring, that will be very costly in terms of the size of the system that you're trying to simulate. And so I fear that this is a level of detail that we will have to go into, to the point of structural plasticity. active dendrites. Really, and then I'm scratching myself behind the ear, like, oh dear, this is really an exascale problem, or maybe even beyond. So I think we're all good natured that we will get there. But I think that the real interesting point will be when we get to that level of detail.
Okay, I think that the conversation you're starting right now is exactly the one that we want to have in this discussion.
Absolutely.
But also an additional part that may not matter to you because you're already convinced, or may not matter to most of us.
Well, I said at this point in time, so there is
okay,
room
no,
for.
Sure, but there are the others still. There are plenty of others out there who will say this is fundamentally impossible, and then they'll come up with some reason. And maybe they happen to have a first name that goes by Miguel, and they'll say it's because everything's analog. Or there'll be someone else with a first name by the name of Stuart or something, and they'll say it's because of microtubules. And you have other first names out there who we can also mention their name completely. I'm just being silly. You know what I mean. There are others who will say this is fundamentally impossible, and so that's why the fundamental discussion about scale separability, that you can even make a cutoff somewhere, that's also an important part of it.
Yeah,
Maybe
I'm
not
also
as
really
important
excited
as
to engage
how
in
to
the
solve the practical
discussion.
problem.
I did want to ask if you could maybe define a little bit more about what you meant by modules, and those modules connected by that interweb.
Yeah, I guess this is just a kind of boxes and arrows view of the brain. I guess you could go as far as to call it a paradigm. I mean, when I think of the brain, the really living biological thing, there is just this incredible, intricate complexity. And I mean, I remember some papers from studies in mice where it turned out that the visual cortex was actually more active when the mouse was doing a motor function than when it was doing a vision task. And so this idealized picture of there is a retina and an LGN and a primary visual cortex and a kind of perfect visual hierarchy, and each of these components performs their certain task, and they can be composed into larger circuits, almost analogous to electrical circuits. And this is also how I first was introduced to the cortex. How does the cortex work? Well, I mean, we're coming to a better understanding gradually. I mean, it's clear that we're also getting a high level view of what these neural circuits are doing. And so maybe that's an interesting point to go into a little bit deeper. Let's say we have a hypothetical robot being. So let's say we want to make a smart home assistant, quite Android-like. Let's say quite similar physique to a human. And could we use this kind of modular approach that I was just describing to make an intelligent agent that will help me do the dishes? I'm sure we could do that. I mean, deep nets are already demonstrating that, well, maybe not quite that, but some great promises in that direction. And I don't think that would be a leap that we wouldn't see happen within our lifetimes or within the next decade. But I guess we at Carbon Copies are fundamentally interested in making an upload of a biological brain. If you don't mind me using the term upload. So it's not that we have the luxury of being able to describe an intelligent agent from scratch and design it from scratch. We are constrained by what we find in this biological organ. And so that implies that we would first have to reduce this intricate complexity that we see in nature to this kind of idealized modular picture. And that is a step that I question.
Thank you. And do you think that the modular approach, because we're giving an example of like a home assistant robot, and where it's you're talking about like motor control and interpretation of its sensory perception of the environment. Do you think that that kind of modular approach can also be worked for aspects such as like consciousness, things that are right now just solely understood from the first-person perspective?
Sure. So yeah, Randall was kind enough to point out this this theory that I hadn't heard about consciousness. So I'm always a little bit wary of the word, but I was very thrilled to read these articles about attention schema theory. And so this is a kind of mechanistic way, if you will, to describe what we typically mean when we say consciousness. In a nutshell, it implies that we as intelligent agents we are constantly building models of the world inside our brain. So we're, you know, if we're playing tennis, then my brain is kind of computing the future trajectory of the tennis ball in order so that I can, you know, swing my arm in the right way and hit it back. And attention schema theory basically takes this one step further and says that we also observe our own actions. And so we also build a model of how we respond in situations. And we build a model of ourselves, essentially, of our responses. And this model, this is accessible in our access consciousness, if you will. So we can think about how we work in the same way as we can think about how the dynamics of the tennis ball works.
Interesting.
Yeah, so I think this is really a beautiful mechanistic way to describe consciousness. Yeah, I'm not sure into what detail we should go. I mean, we recently had great workshops on alternative views. Yeah, integrated information theory and so on.
Right. Yeah, and yeah, it seems like it's portraying consciousness as something that there really isn't anything like superficial about it. That it's like there's a mechanical process to it, and that can all be explained, and also like you know, you could also attribute it to like exactly the same way that like a sensory perception works then, and so.
Well, it's the same neural circuit that is ultimately underlying it. We don't have special consciousness neurons or something like that.
Interesting. Okay. Well, not to go too far into the like the underlying question, but is there any point for in the detail of that, like maybe going back to the modular approach, is there any level of resolution that is like obviously like redundant in the instance of like what is required to emulate as far as achieving the success criteria for, like, let's just say, for example, well, like let's just say the cortex, for example. For everything that we want to emulate in the cortex, that would be for the success criteria for an emulation of that for what we want from it. Like given all of the detail resolution that you mentioned before, is there anything that would be considered, or that you would consider to be a level of detail or level of detail smaller than that, what that would probably not be required to emulate in order to achieve the success criteria that we want? And we'll use the cortex again, for example.
Yeah, so, something that is definitely not necessary.
Correct, yeah.
It's a really interesting question, and I guess that the best answer to give is no definitive answer. We're homing in. I have the feeling we're homing in on the right level of detail. I mean, I remember a time before my next door neighbor knew what epigenetics meant. Now it's in the common vocabulary. That's not such an old invention or discovery, if you will. But epigenetic regulation is, I imagine it's something that would be crucial to simulate the processes that we find in typical neurons in the brain.
If you don't mind my jumping in, I'm very curious. I like the question that Michael just posed, and I was wondering if I can poke you to take it a little bit further. You're thinking about depth or things to include in terms of a higher resolution into the biology. And so you're talking about epigenetics and might talk about molecular processes of some kind. But there are other sorts of resolution as well. So there is temporal resolution, for example. At which temporal resolution do you have to examine and understand what's going on in these brain components? And there is overall scope. So I know we took the cortex as the example, but you might decide, for example, that there are some parts of the brain that don't need to be simulated or emulated in as much detail as others. So it's interesting to know which ones those are. Plus, then there's the whole, do we need to include things that are outside of the brain? Do we need to care about our peripheral nervous system, our second brain in the gut? Do we need to care about the external environment that is handling a lot of the way that we receive signals? And as Steve Potter in his interview said, computing a lot of things for us in that way. Do we need to care about those, or is that just getting a little crazy? And so those are the real questions that I love to hear people's ideas about and their personal views. And also, I think there's this sort of overarching thing where real neuroscientists, such as yourself, always have a tendency to want to be super careful because nobody likes to be wrong, even though being wrong is perfectly normal for us because we get peer-reviewed all the time. So we should be completely comfortable being wrong, but we're not. So we always say things like, the safe thing to do is to assume that we know nothing. But isn't that just a bit of a cop-out? Do we really, at this point, still know nothing about the brain?
No, we know a lot. But it seems like as we have discovered more at this lowest level of detail, at the molecular level, that this has opened up such a world that was not visible before, that maybe we have to revise our assumptions.
Can you give an example of
Yes,
a lower level of detail that has had a big impact on how we understand cognitive processing?
Well, in my master's thesis, I was working on simulating the flow of ions on the neuronal membranes. And so there were some interesting effects that happened because of the space between neurons. it's very active. I mean, there are a lot of neurotransmitters and messengers and things floating around, essentially. Because the space between neurons is very narrow, on the order of nanometers, this results in some very interesting dynamics, some odd diffusion behavior, and it was during this time that I also read about ephaptic coupling. So this is the idea that the concerted activity of large groups of neurons generates an electric field, a macroscopic electric field. And the electric field then kind of bites back on its own tail and results, is kind of sensed and picked up by the neurons again, and it influences their memory potential. So the collective behavior of a large neuronal population has a modulatory effect, and this can be quite significant at the order of a few millivolts, where, you know, to bring the neuron to threshold, you might need 30, 40 millivolts. So this is, and I mean, this is one of these things that is exceptionally hard to simulate. I mean, well,
it's hard to
be
simulate
done
in great detail, right? But we're talking about large
Sure.
populations of neurons. This was
Sure.
actually, not surprisingly, also a topic that came up in the conversation with Steve Potter, Ephaptic coupling, as well as chemical signaling by glial cells and that sort of thing. And the conversation, if I can just clue you into where that was, since he's going to be on the panel workshop as well. It went in the direction where we started talking about, so, okay, what has an ultimate effect when we talk about what are the effectors in the brain? What has the effect on say, the cognitive output? So, for instance, you react to something and you move. And ultimately, because it's the neurons that drive the muscles, it is the effect on the neural circuit that matters. Hence, the ephaptic coupling has an effect on the neural circuit. The chemical transmission by glial cells has an effect on the neural circuit. So yes, it does add extra signaling pathways and types of signaling and ways in which you would simulate that. But it is still an effect that you then translate into I/O for neural circuits, in a sense.
Certainly, yeah. We have tools right now where that allow us to simulate exactly this, and this allowed us to get the estimates of, you know, 1 to 2 millivolts in a typical situation. So yeah, this absolutely. I mean, this is no showstopper. This can be done. But then there is an accumulating list of tiny details that have some functional consequence on the operation of the circuit and the output of the circuit. And so I'm actually very open-minded in that we will be able to build intelligent agents with basically almost the tools that we already have today, and maybe not that much more, but that this is somehow fundamentally different from trying to capture what is going on in the biological brain. Now, an interesting question then pops up. Maybe, I mean, you know, let us maybe take the hypothetical case that we had a perfect brain simulation of a human being. Let's assume we know all the mechanisms. We have a computer the size of the moon, and it's simulating every protein and every little thing. Now let's say that we take the same simulation, but we take away ephaptic coupling. We somehow compute the field and add the reverse of the field, negating its impact. It would be a very interesting question. Well, what would happen to the simulation of that individual? Would they have a subjective experience? Would there be an objectively measurable experience? Would it be worse in NBAC?
Is there any way to test that before emulating? Can you test this in actual subjects?
I suppose you could train networks to perform certain tasks and then handicap them in some way and see how their performance might degrade or not.
I mean, You can even do that with rats, for instance.
Ouch. Yeah, sure.
Simulation, right?
Yeah.
I'm just curious to know, because this is like a fundamental thing that I would give to anyone who says, so they're the ones who have very specific claims, like Stuart Hameroff, who will say, without microtubules, you have no consciousness. Can't work without it. So then I want him to show me in the chain of things that he believes creates consciousness, where does it fit in and why is it essential?
Yeah.
And can you demonstrate that? And you know, the same question goes to the people who are more general and say, I don't know what's really necessary, but there are a ton of little details. Then I always want to know, can you please show me a detail where we've actually tested that difference? Because it's not, I'm not saying that there is no difference. It's just that by seeing what the difference is, what a difference it makes, you can often discover what it would take to replace that if you wanted to do a simulation, or how you would go about, say, doing what you did, take the Ephaptic coupling away, but then perhaps somehow incorporate it still in the equations through some other way.
Yeah, have a functional equivalent.
Yeah.
That is maybe computationally less intensive.
Right.
Yeah.
And you said that we have the tools in order to model all these things. I just for clarity, are we still just talking about things like writing a code for part of a simulation, or are we talking about like actually replicating that electromagnetic field, like as an example?
I think that was specifically about the ephaptic field. Yeah, there was some great work done on this. I think it was one of Christoph Koch's PhD students around the year 2000. I'm really bad with names. I'm also thinking in terms of code, I'm thinking of the neuron simulator and a tool called LFPy, which work in tandem to have just this interaction of the field with the neurons.
Okay. It's actually, there are also, of course, those who claim that you cannot do this through simulation, that you have to build things. I had a long conversation with an Australian researcher who, a long time ago, many years ago, who claimed that because simulated fire doesn't burn, you cannot ever produce thinking minds and consciousness if you're just using software. He didn't even go to IIT, he was just straight up like that.
That reminds me of my old neurophilosophy professor with whom I had a debate about whether simulated pain can be real. So needless to say, we were on different sides of the debate. You brought up an interesting subject earlier. I had to think of the knockout mouse. So as you know, there are genetic tools that we can use to knock out specific genes coding for specific, let's say, parts of a receptor. We can knock in to make, let's say, a receptor with different subunits and suddenly different behavior. And then typically what is done is then these mice are put through some behavioral test paradigm. They have to run through a maze and then find some hidden food, or they have to find a hidden platform underwater. And it is then typically in these papers you then see some mild but statistically significant effect on behavior. Behavior is typically worse if you mess something up. So I guess that's kind of good news for they were at some local optimum at least. But one of the things I wonder. So I mean, brains are pretty robust, and typically we can wreak some pretty serious havoc, and the animal will still be pretty okay and be able to find food and somehow produce offspring. But they might do a little bit worse on these behavioral tasks. And so this then is maybe, is this related to the level of detail question? Because I'm wondering if we will have a lot of details that we can simulate or not. and make that decision, which might have very subtle effects on the individual subjective perception, or somehow objectively on simple or complex behavioral tasks. Hopefully we'll not put the humans through the Morris water maze.
We should actually try
So, yeah, sure. So I'm very interested in how we're going to make those trade-offs, and maybe I might even, and here again, like you mentioned, I'm a typical academic, not willing to make any commitments. So I'm hoping that maybe we can even leave some of these decisions to the individuals who are being simulated. So that some people might make different trade-offs for themselves.
If the tools are available to try to go up and down the scale, yes, then you could do that.
The tools and of course the dollar prize next to it.
But if you had the tools to go to the highest possible resolution that you could possibly imagine was possible or necessary, then wouldn't it be more ethically correct to simply emulate the person at the absolute highest level, and then to start testing by keeping their very high level backup, and then doing lower level versions and seeing if it feels any different, comparing reports, doing little, just going lower level on this part of the brain and then that part of the brain, and that sort of thing. I meant by lower level, I mean lower resolution. But the question that you are now going towards is the question of success criteria for whole brain emulation. And that, of course, is the underlying most important thing, because whenever you say, can we leave this out? Question is always based on what is your goal? What are you trying to achieve? And you're right about robustness. The other thing about scale separation and scale and resolution is also how many neurons do you need to sample, for instance? How many do you need to reconstruct? Is it okay if you grab every 10th? That sort of thing. In some brain regions, that may be fine, whereas in others, that may be catastrophic. But that matters, because sometimes people will find it totally acceptable if their memory of exactly what the weather was like 3 weeks ago isn't so great, sure. But if you took away all of their NMDA transmitters, well, that might be a problem. Or if you, just suddenly they couldn't remember any names at all, that would be a problem too. So there are different kinds of loss that are more or less acceptable, and that's definitely a discussion to be had. Anyway, I'm going to sit back again for a bit.
Yeah, no, absolutely. I was thinking maybe the hypothetical case of starting with the perfect emulation, and then gradually decreasing the resolution to see how coarse we could make it. I guess that would be the least ethical thing to do. At least when we're constructing it, we have the excuse that, we're treading in unknown territory.
Is it not ethical if it's a volunteer and they say, I'm fine with this as long as you can always revert me back to my high resolution version?
I mean, people undergo TMS experiments, right? Where like some part of their brain is temporarily disabled by a huge magnetic shock. So I guess if that passes the ethical bar, then so could the other thing.
As far as the challenge is concerned, to like making a precise simulation compared to just an imprecise or like a partial. As far as like the time and the effort, and I guess even the funding involved to do that, it could be like a worthwhile project to first, though, go with an imprecise emulation, especially if we are just not even talking about humans yet. If we're just even maybe even just like a Drosophila, for example. I guess what I'm asking is there would it make sense then in that case if we wanted to have an imprecise emulation first, or well, and also if that would be the case, is there like a kind of a logical path to go about that, like maybe an emulation where there was no memory? I don't know if that's a good example, but I guess another way of asking is there maybe a aspect of success criteria that we know is probably going to take the most time to figure out. Well, if we maybe skip that for the moment, if we just add everything else, then that would be an accomplishment in and of itself, and then we can build off of that. Is that like a practical way to go about it, or does that even make sense?
Yeah, thanks for asking that. So I'm really impressed with the OpenWorm project. So this is a community of people who are trying to simulate tiny little worm with 302 neurons exact. And not only the worm itself, but also simulate the environment. So they have a complete fluid dynamic simulation such that the worm can wiggle around and actually push off against the fluid. So it's typically in water. Push off and explore the environment and forage for food and so on. So it's really cutting out a little part of real life and putting that into a simulation. So this worm, C. elegans, has 302 neurons. The human brain has something to the power 10 to the power 12. And you know, it's not easy to simulate a worm. This is hard enough as it is. C. elegans neurons have some special properties. I think most of them are not typical spiking neurons as we see in most of the cortex, but they kind of communicate with a whole barrage of neurotransmitters, also mostly subthreshold, so without emitting any action potentials. And I think so. It's hard enough, but they're making really impressive progress, and they are pioneers in some way. It seems like it's not going to be like in that science fiction book. So the first uploaded creatures will probably be worms. And yeah, so we're not going to jump straight into emulating the human brain. We are, of course, in parallel. There are many efforts dedicated to doing just that. So there are many problems that you have in emulating the human brain that do not even occur in the case of the worm. So some really impressive setups, for example, have been built in order to automate the process of slicing the brain and doing an automated electron microscopy and three-dimensional reconstruction on that. And this is really impressive because this was kind of feasible, I guess, at the level of the worm, because you would maybe, you would put some PhD students to work on that, and one of them would man the slicing, and one would man the EM, and this is at a scale where we could do this manually, but this would never scale. I mean, there aren't enough PhD students in the world to do that for the human brain, so something fundamentally different is needed there. So this is being done in parallel, fortunately, and it's looking really good. But in terms of actually, and okay, this is maybe an interesting point. So an interesting part of the OpenWorm project is that they're putting a lot of emphasis also on the simulated environment. So they place a lot of emphasis on this notion of embodiment and embodied cognition. In humans, there are also many discussions about this. So the notion that our body is really doing a lot of computation by itself, even in a kind of pre-brain kind of way. The body is its own dynamical system. So it will be absolutely essential that we simulate some kind of body. We cannot be disembodied brains in a vet. I mean, our brains can be in a vet somewhere, but they will have to be hooked up to an interface that will give us the perception of walking in a world or flying in a world or some more exotic things. Sorry, Rendell?
Brain in a vat makes me think of, I mean, some people think of The Simpsons, but I think of RoboCop because they just put brains inside robots.
I was thinking of Daniel Dennett.
Yeah, and I did want to ask a question about the images. This is kind of going off a little bit of a tangent, but because we're also talking about like testing, especially when we're like simulating environment. Is there anything that you would say about just the images, like as far as the images from a slice and scan? What from those can we not? What information from those can we not gain that require like what information can we not gain just from the images that we will need to be able to incorporate into our modeling, perhaps things that have to do with like the temp, like how the brain grows over time. Would we have to know like the way that the brain grew in the image that we were looking at, I guess also is there yeah, well, let me stop that question there.
So when you say image, then you mean, for example, the image stack that we might get from our electron microscopy setup?
Yes.
Yeah. Yeah, this is definitely defining some minimum. I mean, it gets to the point, I guess, where we can decipher the strength of a synapse based looking purely at the images by inspecting the number of vesicles and the density of the postsynaptic density. So we could infer a great degree of the connectivity from this data. But then, yeah, is that enough? It's a snapshot in time. So we talked earlier about epigenetics. So epigenetics would definitely require some reading out of the epigenome. But yeah, and so now it really shows that I'm by no means an expert in microbiology. But maybe so if there are any microbiologists in the audience, then please fill me in. But I could imagine that there would be combined approaches where this EM approach would remain in place. That would be our method for determining the whole brain connectivity. But in addition, we would definitely need some extra procedures to maybe sample the epigenome in different parts of the brain. And then the thing I'm not sure about is how representative that would be for the neighboring area. So what would be the density at which we would have to sample the epigenome or other, I don't know, phosphorylation states in synapses. Receptor densities, how distribution of receptors on dendrites and ion channels on dendrites. So these are complementary things. And yeah, I'm very interested in the discussion with people who are experts in microbiology, who can maybe fill me in a bit more there.
Thank you. Okay. Well, I do have one question about that I want your input on about is about the temporal question again about like we're talking about getting we're talking about emulating the precision of the timing of say like a train of as I understand it, like the like for example train a neuron spike sequence, like if you would give it, for example, if you're just talking about one specific cognitive process, as I understand it in the brain, the spike timing determines, or at least factors into the ordering of the neuron spike sequences, and in turn generates that process of the information. But when you're talking about emulating, when you're talking about emulating those spike patterns, in a computer, like those can be much faster, and so we're able to control like the timing if that happens. But is it possible that when you're emulating the spiking of neurons, meaning like the sequence of how things happen, or how long those spikings going on for, like, and still using the example of one specific cognitive function, could it be sufficient to just emulate the sequence of when that spiking happens, rather than trying to get the exact timing right? So in other words, what I'm trying to say is, when you're talking about the resolution of the timing for a sequence of neuron spikes, if you just want to emulate if what your criteria is emulating the information that was processed in that one cognitive, in that like specific cognitive function, how important is getting the timing right for every neuron spike, or is it just a simple matter of getting the sequence correct, like this spike happens before this one, and this one has to happen before this one finishes, kind of like with parallel computing, where you just have to simply get the order correct.
If we're simulating the spikes coming out of one axon, don't they always come in the same order? Or are we talking about the spikes from different neurons?
The spikes from different neurons.
Okay.
Timing is really important. So yeah, in terms of the resolution question, ideally, we would not have to simulate the precise shape of the action potential. It would be much more economical, computationally speaking, if we could summarize, if you will, an action potential by just a discrete event in time that has one time step, one floating point value, and that's all we need to describe that spike. If you have a full Hodgkin-Huxley model running for your neuron, then whenever it produces a spike, it's going to actually exhibit very precisely that action potential in the dynamics of its membrane potential. And well, I guess we're lucky in the sense that neurons are comparatively slow to computers. So our networking gear that connects the different nodes of our parallel distribution simulation together can have maybe, you know, a few dozens of microseconds of lag, maybe even a few hundred of microseconds. And this at my left, where they evaluated in a comparatively simple network what the impact was of choosing a time step and aliasing spikes to that time step. So the thing is that, let's say, you know, you have your model, your brain model. It's formulated in the form of a dynamical system. So in terms of dynamical equations, you simulate these equations on a computer. It's kind of, you know, there's an analog dynamical system. You're simulating it on a digital computer. So you have to discretize things, and you have to choose a finite resolution, a finite time resolution for your simulation. So you kind of, you know, make jumps in time with a discrete step. And typically you would choose something on the order of maybe 1 millisecond or 0.1 millisecond. And now the trouble is that it does seem that timing is really important with neurons. You know that there's one of the plasticity models that is exhibited in the cortex is spike time independent plasticity. So this is the phenomenon where you have a synapse connecting two neurons, and the presynaptic neuron could fire a spike, and the postsynaptic neuron can fire a spike. And if the presynaptic neuron fires a spike before the postsynaptic neuron, then it seems there was some causal connection. The presynaptic neuron fired, contributed to the firing of the postsynaptic neuron, and so that should then result in a change in the way that the synapse, a change in the strength of the synapse. But the trouble is that
if I pop up an image of STTP.
Yes, please. Is it going to be the STTP curve?
Possible image, but it's nice. It's nice to be able to do something like that since we're in this medium. So it's going to pick a window here. I think it would be this one. Hopefully this is the right one. I'm not sure if I'm sharing the right window.
Ah, fantastic.
Is it there? Okay. Good. So now you can talk about it while it's there.
Yeah, that's really good. So we're exploring more possibilities of the medium. Yeah, so I mean, what it comes down to is that the STDP. So STDP is what we're looking at essentially is a model of what we find in the real brain. So we have particular combinations of presynaptic neurons that are connected via synapse to a postsynaptic neuron. We stimulate them with certain patterns, pre before post before pre, in all manner of different combinations. And we look at how that affects the strength of the synapse, which is what you see at the vertical axis. Now, there are different kinds of STDP, and they're defined by these different rules, if you will. So what you see in these particular instances of STDP is that there is this inversion that happens around t equals 0. So when the presynaptic spike is happening almost at the same time as the postsynaptic spike, we're kind of in an uncertain region, whether we get a very strong depression, so a weakening of the synapse, or a facilitation, a strengthening of the synapse. So this phenomenon is really sensitive to the precise time of the spikes that arrive and that are generated. So a consequence then that my colleagues found is when you choose your time step too big in your simulation, then spikes can only happen at increments of this resolution, because this is like the resolution of your universe that you're simulating. There's no smaller time than the time step that you're simulating it. And what happens then is that a spike that might, in the real universe, which is analog, would have happened between two time steps, is actually aliased. It's rounded off, so to speak, to the edge of a time step. And this induces an artificial synchronization mechanism. So actually, informally speaking, this makes epileptic brains, because you get additional synchronization. And sometimes this can mean that your whole network becomes completely synchronized and it all fires at once. And then you have simulated epilepsy.
Of course, this is especially a problem when you're talking about an unconstrained, highly nonlinear analog system.
Like a neural network.
Like a neural network that is unconstrained. The brain is not an unconstrained neural network. You know, I mean, this is all very different when you're applying these same rules within a network that, for example, is giving itself a theta rhythm, that is giving itself recurrent inhibition to produce gamma rhythms that will make sure you don't get a whole bunch of other spikes occurring right after your pattern fired. So these things do matter. The constraints really matter. Without the constraints, I think that someone like our friend Miguel, who believes that the analog system could never be modeled because you've got this chaotic connection all the way from the lowest level at the smallest time scales up to the large macro scale because of these aliasing problems, would have a point and could say, you're just never going to get right. But then when you look at practical implementations, such as the hippocampal prosthesis and how they're modeling it, you'll see that they can get away with an awful lot of abstraction and still have a positive effect, at least in a support role for that brain region. Because it doesn't even matter that you get the spikes to happen at exactly the right time, as long as you're modeling the membrane potential, and you then put some stochasticity on top of it so that your spikes are happening around the time that you would expect them to happen, since they're not perfectly predictable in real neurons either.
Could ask, I was curious, as far as what you thought, like with the modeling experience that you've had, if there is any redundancy that you've noticed that wouldn't necessarily have to be modeled for every individual in particular, but could be generalized across individuals, making the process a little bit quicker, I suppose, like neural types or even whole regions of the brain. If it's possible to even have a database of these things that we wouldn't have to have that detail for every single individual because everybody shares it.
Yeah, so again, I'm not a biologist, but I suppose there has to be a huge variation in brains. I mean, I guess we've all seen the case of the person whose ventricles were blocked at an early age and who then had a head filled with mostly water and was getting by apparently fine in their life. So yeah, it's really, the question is really related to the level of detail question. Because if we can understand the brain at a high level of abstraction, where we can have a V1 module representing occipital cortex and so on, and these could be shared in a database. Man, we're pretty close to the matrix at that point, aren't we? Yeah, I mean, that would be really great, but right now, I guess from my conservative neuroscientist point of view, the most reliable prediction would be that we would have a fairly data-driven pipeline, which would begin at this sectioning and scanning of individual brains, and then to use a generic infrastructure to simulate them. So we wouldn't have to reinvent the simulator and the way that we analyze the data and so on to construct the model from the data. This, I imagine, could be shared to a very large degree. But the individual brains, well, I guess that's the really cool part that we, yeah, tricky and cool.
Well, I guess what goes along with that question, though, is so we've talked about a little bit about electron microscopy, but that gets us to like the connectomics level. And if there are identifiable neural types or maybe a better way to put it, if there are things that we observe perhaps at a greater resolution but can then match to what we can learn at a connectomics level, like, do you think that that's going to be possible, that there's enough redundancy there that we can identify what's necessary from a connectome to be able to then create an emulation?
So you mean if we were to analyze a lot of connectomes and then analyze what would be the commonalities between them?
Not just that, but even if we had data, like we had research at a higher level of resolution or at a greater resolution, to be able to identify patterns, I suppose you could say. So if we would be able to identify, oh, okay, we can recognize this in this neural type in a connectome, and we know based on what we see here, because of other research, what to expect at a lower resolution. So what I'm saying is, do you think that this is going to be done in two parts? Like for emulation of an individual themselves, we may need at one resolution, but in order to emulate that person correctly, we'll need to understand a different resolution, or at least have data from a different resolution.
Well, So I'm not sure. The only thing that came to mind was that the components of the brain, if you will, the neurons themselves, the channels, the neurotransmitters, on this level, we share a great deal. This is quite conserved across individuals. Heck, we're pretty close to mice in that respect. So yeah, then I mean, I'm myself working with a database of individual neuron models, single cell models. So I would be very much in favor if we could compose simulations of brains using those models as a template. So yeah, maybe that's getting quite interesting. So let's say that from our EM scan, we are able to identify that a certain neuron is of a certain class. And don't get me started about classifying neurons, because I'm not a strong believer in that either. But let's say, for the sake of argument, that we could say, okay, here's a pyramidal L5 neuron from this and this region of the cortex, from your primary motor cortex. We look in our database, and we find a highly detailed neuron model, a dynamical model that we can simulate of exactly that class of neuron. So now we suddenly don't have to do the full, let's say, scanning of where are all the channels in the active dendrite, because we already did that in a separate experiment, and this has been entered into the database, so we can use that neuron as a template. That would be really cool.
Do you have any expectations along that train of thought based on what you've seen so far?
I'm not going to say it can be done. I mean, it probably will be done. I suppose that, you know, this is science. We have a lot of international teams who are working on this problem. Actually, I don't really have a good overview of what, for example, is happening in China. I'm sure there's a lot of research being carried out that would serve towards getting us closer to whole brain emulation, although maybe not under that name. But yeah, I mean, I'm sure there will be a great diversity of approaches. There's some really esoteric stuff, like one of the proposals that I came across was to have tagging of individual cells. So you would have some, you know, genetic trick out of the magic head of the microbiologist, and they would allow kind of a complete chimera state of the brain. So every cell could be individually tagged with a unique code, a unique DNA code, which we could then later somehow read off, or we could have the cellular factors. So for example, the state at a synapse, determining then incorporations into the DNA and reading cells out like that. I don't know, maybe there's a lot that we can learn from that. So yeah, just a lot of different approaches, and I guess somehow, you know, through all the chaos, some really cool things will emerge.
I guess going back to you mentioned about epigenetics, and if you're looking at the epigenetic or simply genetic level of an individual neuron or group of neurons, are there certain predictable behaviors?
Sure. I mean, yeah, at the very base distinction between excitatory and inhibitory cells. I mean, so maybe this is actually getting into the discussion of, can we classify neurons? And maybe again, this is a cop-out, because at some levels we certainly can, like the excitatory inhibitory cells. There's a lot of conserved behavior, even from mice to men, looking at, you know, the same types of cells in the same position, same layer in the cortex. There are quite some similarities, and I know we also know about the differences, but So there's certainly classification that can be made, but the classification again works at a certain level of description. Excitatory and inhibitory are the broadest classes that you could imagine and cover an incredible diversity. So it's more like a hierarchy of classification than a final being able to put one neuron into one box and another into another.
Okay. Interesting.
But yeah, in a practical sense, I'm sure we can get a very long way by having your standard dentate gyrus neuron and so on, and putting that together.
I did want to ask, is there anything that we did not cover that you would like to say to our audience about the topic, or anything that we covered in the questions?
Yeah, I was thinking, so there was this somewhat older paper from 2011 that is about how to write a simulator, essentially, and in a way that scales very well to very big brains. It's written by James Kozlowski and John Wagner when working at IBM, and they called it the Neural Tissue Simulator. And this kind of ties in with the question of level of detail. So one of the questions that come up in simulating brains is you have a certain model, a dynamical system, which could be really big. I mean, you might be simulating, 10 to the 12 neurons. How do you spread the load over multiprocessing system, over a parallel computing system? So there have been various proposals. This could also be something that's very data-driven, so we could inform that decision by the connectivity of the brain that we're trying to simulate, or connectivity of the network that we're trying to simulate. But the proposal by these two gentlemen here is that we really take into precise account the spatiality, if you will, of the brain. The brain is an object which has, an extent. It's like 15 centimeters by 10 centimeters by something. And they propose to make this distribution along these Cartesian axes of the brain. So we divide the brain up into volume blocks, and these would be equal size volumes of tissue that would be adjacent in space, in simulated space and in real space. And this would inform then the network topology of the computer that we're simulating it on. So because any volume of space can only talk with adjacent volumes, this means that network connectivity is completely local. And this avoids a lot of the communication. So a lot of the problems in neural network simulation is not so much simulating the neurons, it's the communication. There's a lot of spikes that have to be transmitted over some kind of network connection. So I still find the approach this approach by the neural tissue simulator very elegant, and I think that will scale really well. So this might be a real good candidate.
Yeah, I was
for the
really
long term.
attracted to that model as well. Actually, I went out in, I think it was 2013 or 14. I went to New York and I visited James Kozlowski at IBM.
Oh, wonderful.
Talked with him about the model, and it was really fascinating. I do think it's a very interesting approach, at least to the problem of how would you run an emulation like that once you have it. Unfortunately, I haven't heard very much from him since. I think the last time we were communicating was around the time when that Global Futures Congress was being organized. haven't heard much from him since, and I think he hasn't continued on the same project because for IBM this was sort of a sidetrack. Yeah, but yeah, that's super interesting. I have to actually go, so I just wanted to thank you before I rush off and they can finish. One topic that I often also like to bring in as far as making things more difficult is that it's not even just what do you need from the brain and how do you build a simulator that can handle simulating the brain, but the fact that even bigger issue than, say, communication between neurons is tuning parameters, because machine learning always explodes into infinite numbers.
Absolutely.
So that's the big one. Anyway, I have to run, so I'm very sorry about that.
Thanks very much. It was very nice chatting with you.
So I will now log off.
Hope to follow up soon.
Thank you, Randall, and thank you, Charles. I think with that, you can call it an end for the interview. So thank you very much.
Yeah, thank you so much for putting this together. I'm really looking forward to the workshop. I'm very excited about it.
Thank you. Okay.
Thank you so much.
Many thanks to Dr. Charles Linson for that great interview. We transition now to our live panel discussion, which is introduced through a keynote lecture by Dr. Ken Hayworth. Here are just a few reminders regarding how the discussion will go. Our workshop is participatory and is open to all of our international attendees and viewers. You can write questions directly into the chat that is enabled for our YouTube livestream chat. One of our excellent staff members is monitoring the chat and will alert the panel to interesting and topical questions. More complicated or multi-part questions, where it is important to establish that the question is both properly understood and answered, are best asked by directly engaging with the panelists. We make this possible through our call-in number. The number and PIN are shown at the bottom of the screen right now. They're also written in the live stream description and on the livestream.carboncopies.org webpage. They are as follows: 786-540-5631 with PIN 180-618-986 symbol. If you're calling from outside the US, you may also use the web call-in option at call.carboncopies.org to avoid telephone charges. The process is similar to calling into a radio show. Your call will be monitored and a respectful exchange is expected. When your question would benefit from a direct interaction, I will invite you to call in if you can. If you want to add a note to your question stating that you can call in or that you cannot, that can also be very helpful for a moderator. First, we will play a short clip introducing Dr. Ken Hayworth.
Dr. Kenneth Hayworth is a senior scientist at the HHMI's Junelia Farm Research Campus and is president and co-founder of the Brain Preservation Foundation. Dr. Hayworth is exploring ways to extend focused ion beam scanning electron microscopy imaging of brain tissue to the volumes of mammalian and human brains. While a postdoctoral researcher at Harvard University, Dr. Hayworth co-invented the tape-to-SEM process for high-throughput volume imaging of neural circuits at the nanometer scale. Dr. Hayworth is at the forefront of efforts towards brain preservation and whole brain emulation. Dr. Kenneth Hayworth will be speaking on scale separability.
We'll now begin the panel discussion with some prepared questions for Dr. Ken Hayworth. Dr. Kuna, would you like to give the first question to Dr. Hayworth?
Oh, is that how we're starting? I wasn't sure if I was going to ask
with questions
first or if Ken was just going to start talking.
Oh, so he would have the lecture first.
Well, I was told this was an interview, so don't expect a lecture.
It kind of is. So I just assumed that you'd have something because you typically have like a bunch of slides, even if we say, oh, we're just going to do an interview.
I mean, I will say I listened to the first two speakers, and I completely agree with them. I think there is something that is a level that neuroscience is assuming is the correct level for emulating the brain, for asking these questions. And that level is, for want of a better term, spikes. Is the electrophysiology. Let me give a little. I was thinking about these questions a little bit, and let me give a little bit of a different take on it from maybe my computer science background, because I've as a bachelor's degree I was going through computer engineering. Really understand how to put together a von Neumann architecture computer, and so if you think about computation of the brain, what you're going to want to think about is you're going to want to think about representations, and you're going to want to think about processing computation on top of those representations. And so the representations in the brain have to be able to be transmitted across the entire brain very quickly and efficiently. Otherwise, you won't be able to get the entire brain involved in certain computations. And so that really limits us to the types of things that can transmit information efficiently across the entire brain. And obviously, as neuroscientists, we recognize that that is action potentials. And so it should be clear that the representations that are at the heart of all of our cognitive processes are going to be groups of neurons shouting action potentials to other groups of neurons way across the brain. Now, that just talks about the representations themselves, but now what about the processing? Well, the processing could be any order of things, but it has to quickly assimilate the spike trains that are coming from different areas of the brain and do whatever it's going to do with those, and then spit them out again as spike trains. Otherwise, it wouldn't be useful. So, so if you're talking about, let's say, a chemical process that might change messenger RNA expression, et cetera, that is going to simply take too long, I would suggest, to interface with the spike trains coming in and to give spike trains going out. So what we have is the processing. We've identified this as neurosciences, I suppose, as being involved with ion channels, receptor proteins, things that can immediately take in these action potentials that are releasing, let's say, glutamate across the synaptic cleft. That glutamate is almost immediately, within a fraction of a millisecond, turned into another voltage that is sensed by voltage sensing ion channels, and that is making it very efficient to accumulate those and either put out a spike or not put out a spike. I'm saying stuff that any neuroscience textbook would have in like the first chapter, but there's a reason that this level of computation is the one that stands out as the one for whole brain emulation, because it is efficient in transmitting things across the brain, and it's efficient in it's essentially that it's a representation that is efficient in transmitting things across the brain, which allows many areas of the brain, many variables to interact collectively.
Right. Yeah. So that addresses the question of the forms of communication, the kinds of channels that we need to care about, because we could say, as long as we can translate them into the effect on a membrane potential, and therefore when a spike is going to happen, then you've got your bases covered. That still leaves other dynamics such as memory, memory formation. And yeah, I'm not going to put words in your mouth, so I'll just ask you, what's your take on that, and on, say, the factors or the level, the resolution at which we should care about contributors to that memory process, all these different stages from short-term to LTP to long-term and so forth?
Yeah, well, I mean, let me first say that unlike the first two speakers, I have very little expertise in simulating neurons. I've written a couple little papers that hardly anybody reads that have something to do with neural simulation, but very little. Where I'm coming from this really is, I think, the overall question of this, which is what is the level of separability? That level of separability is very important from a brain preservation point of view, because if we don't know what that level of separability is, if we don't know what the level that is that the mind is running at, then we cannot say what would be necessary to preserve in a brain. And so I think deeply about these questions because mainly from that perspective. And from that perspective, you come up with things like you're going to want to preserve all the neuronal connectivity. You're going to want to preserve the ultrastructure of dendritic spines, for example, so that you can get their volumes, their area of postsynaptic density and number of vesicles and things like that. You're going to want to preserve, in the best case, you would want to preserve all the ion channels, all the receptor proteins. You would want to preserve potentially their phosphorylation states, et cetera, et cetera. Messenger RNA expressions, local messenger RNA, all of these things. And the good news is that we can preserve all of those things now. But the question of separability is really important because people tend to agree what level that is until you start to talk about the question of mind uploading. And then you get to, wow, maybe there's even something lower than that and lower than that. And to really address those questions, I think you have to go into our deeper theories, our cognitive theories of what consciousness is, what intelligence is, what memory is. So with that, I think I've definitely lost the question here. So
Don't worry about that.
Repeat the question.
Yeah, but don't worry, we're going to get back to it anyway. In fact, I think that this is a great time to also open up the floor to everyone else who's here, because I really don't want to leave the other guests who joined us, like Dong Song and Yoon-Suk Cho, and of course Steve Potter and Charles and others from, they want to have their say too. But I think that for all of us, because we haven't really, most of us don't go out there and debate whole brain emulation with other neuroscientists, which is something that you have done. And I don't want to put you on the spot, so I don't want to say name any names unless you feel like it, because hey, I'm open to it. But if you could give us some idea, since you already kind of pointed to it, what it's like to talk to a neuroscientist about whole brain emulation and where they kind of look for, I would almost call it the excuse as to why it would be impractical. Maybe impractical is even too soft a word. Maybe they say impossible. Could you say a little bit about that?
Yeah, I mean, that's a great question. Let me give you a very recent Kenneth Miller. He was interviewed for something that I was in for CNET cryonics, something. And he was giving his opinion on why mind uploading will not occur in, it will take, I think he said thousands of years. I think that was the thousands of years. That's fine. Okay, but he offered up his explanation of why, right? And his explanation of why was that there was I very much appreciate that neuroscientists like him and other neuroscientists that really know what they're doing engage in this conversation. So, I would recommend you getting him as on an interview to really get the guts, because I've had debates with him that are I've learned a lot. Right?
It's a great idea.
Yeah. In any case, he stressed that there is level after level after level after level after level after level that you have to go down to really encompass all of the computation that is going on. And it was directly this levels of separability. He was essentially saying, no, there is no electrophysiology level that you could draw a line at and then say you model everything else with some local differential equations, which is essentially what we're talking about, right? I mean, we're talking about obviously it requires proteins to do the work of the Hodgkin-Huxley equations. But do you need to model the protein conformation in order to get the level that you need, right? And I think we would say that that's as silly as saying if you wanted to emulate a computer that you would have to know the exact locations of every dopant atom in the silicon crystal of the transistors. Obviously, you have to have transistors and you have to have dopant atoms in those transistors, but you don't care where they are. In fact, there are many, many, many levels of abstraction that you can go to before you really hit the level of computation. And so there, this whole question of level of separability really is the thing that people hit me with.
Actually, Yunsuk, you could just jump right in right now. I think it's a great time to start talking directly between all of us. Oh, his mic is not working. In that case, I'll present his question. Yeah, when one is considering the separation of the levels or scales, doesn't one also need to consider how those separate levels will interface or interact?
I don't think so. Can you give me a more of a idea of what two levels you're talking about? So I can, instead of imagining two levels, if
I'll
you
wait
can
for
give me...
Yunsuk to put in his answer to that. And in the meantime, I'll give my version of your answer, which is the whole point of scale separation, of saying there is scale separation, that you're separating scales, is to say that you have determined that for the context, for the goal that you're emulating towards, let's say we're not trying to emulate to understand how Parkinson's disease works. We're trying to emulate how it is that a memory pops into my mind. If you have your goal set and you determine you've found a level where you can separate scales, it means that above that scale, at those resolutions, that's what you have to care about. That's what you're emulating. It's like the code of the program that you need to emulate how it's being run on your emulated Macintosh. Below that level, you don't need to care. We don't need to care about how the Macintosh implements its chipset. That's the idea. So the answer to, would you need to care about how they interface then, would be no, as long as you believe that scale separation is possible.
So maybe also something that connects to that is that one of the problems, if we were to do our simulation at a high level of abstraction, at this kind of boxes and arrows level that deep neural networks are currently operating at, then we would need to have a complete understanding of how the brain works at that level before we could replicate that in a simulation. So we'd have to know, the age-old question about how do what is neural coding? How do neural codes work? How is information encoded into a spike train? The rate coding versus precise timing of spikes. If we were to address the problem at a smaller level of detail, so looking more towards the individual neurons and individual proteins, then we kind of sidestep this question. We just simulate the system in a data-driven way. We kind of take what's given to us, and we don't have to understand exactly what is then encoded in a spike train.
I would love to jump in on that one, Charles. Because, okay, so this is kind of getting to the fundamentals of whole brain emulation. When we first got to talking about this, maybe back in 1994 or something like that, our optimistic young selves would say things like, well, whole brain emulation is a fantastic idea because we don't need to know everything about the brain to make it work. Because as long as we understand how neurons and synapses work and we can scan them all in, then we can make something that works. Of course, ourselves at that time were not yet very familiar with things like machine learning and model fitting. So we didn't yet appreciate that, first of all, if you're going to do that, you would need to have the data for all of those parts. Now, if you can just do this by scanning, by reconstructing from scans from EM, that gives you one side of the problem. The other side of it is what Dongsong probably encounters when building hippocampal models, is that once you are building your model, the more dimensions you have, the more channels, the bigger your model fitting problem gets, to the point where very quickly it says something on your computer like, okay, I predict that this is only going to take 10 to the power 1,000 seconds to compute. But 10 to the 1,000 seconds is, more time than there is in the universe. So that's a problem. And that's kind of what the naive version of looking at whole brain emulation bumps into. Leaving that there.
So would you say that this is not sufficiently constrained by the data? So we have data at the electrophysiological level. We can measure spiking activity going on in networks, but we cannot directly infer what the neural code is from that. But if we look at a smaller scale, then maybe it's easier to replicate the behavior at that scale and validate that we're doing it right, without indeed developing a full understanding.
Well, in principle, what you're saying is true. The smaller the system that you're trying to do, that you're trying to model, the more feasible your problem is. So if we know enough about the brain that we can say, we have a blueprint here that tells us that prefrontal cortex does this, and it should be exhibiting these kinds of rhythms, etc. And in the prefrontal cortex, we have a column structure that looks like this, and it's got these types of neurons, and typically they operate like this. And then you have your actual recorded data from a patient, whether it's electrophysiology or it's EM. And you're mapping this to the blueprint that you've got. Then perhaps you only need to look at and examine what's going on in a small circuit that is a part of a mini column in the prefrontal cortex. And you're trying to tune that. And Dongsong, you can correct me if I'm wrong, but in a sense, this is what's going on in your hippocampal model, right? You know a lot about the underlying blueprint of CA3 to CA1. You know a lot about the responses that you normally see from those neurons. Mm-hmm. You have a very constrained experiment in which this is taking place. So it's not a very rich context. And within that, you can then make a model that will actually come to some kind of conclusion within, 72 hours while the patient's recovering. Is that right?
Yeah, I mean, when you look at the model, right, although the model I'm developing, I call that also a data-driven model, but there's a lot of assumptions and knowledge already embedded in that model structure. So that's already reduced the dimensionality of the problem. And also there's advanced model estimation method like the dropout estimation, like regularized estimation, that further reduce the complexity of the problem. So you can use limited data to solve a complex system, right? That's very common in machine learning now, right? Some machine learning algorithm, they have like millions of open parameters, but they didn't use over a million images to train those model, right? When I was a graduate student, I didn't even think this is possible because obviously, right, you cannot have the, my understanding at the time is that the number of unknown parameters has to be, right, smaller than the number of the samples, right? Because you have, otherwise you have a, ill-conditioned problem will never give you a unique solution, right? But now our understanding changed because there's a lot of constraints in the model structure. There's a lot of constraints embedded in the estimation procedure. So you are not looking for arbitrary solution for given that data, right? So in the end, those problems actually it's tractable in reality, right? And another thing, I agree with you. It's when you think about problem, right? Like input-output model, you try to map input from output, but you are not really trying to estimate that input-output mapping for arbitrary input, because in reality, the input pattern is also limited, right? Because I'll give you one example, trivial example, that if you only want to simulate one spike, right? Obviously, all the scale separability problem is already solved because you send one spike as your input, right? You only try to simulate the, for example, like EPSP as your output. You can easily just copy that EPSP waveform and pass to the next neuron, right? But this in practice this is not very useful because we are interested in spatial temporal patterns spikes. We are interested in cognitive process, right? We are interested in behavior. Usually involves a lot of neurons, a lot of spikes, a lot of EPSP, right? But in reality, you also don't have to solve all the problems at once, right? If you can find some scenario that make your input-output prediction useful, then that's by definition is useful, right? My favorite example in, it's really hard to talk about this in terms of memory prosthesis because it's more subtle and it involves more complicated behavioral tasks. But my favorite example is like in retinal prosthesis, right? So if you read about retinal prosthesis, the current system is extremely crude, right? I remember 20 years ago, we talked about 4-to-4 array of electrodes. So basically, you are representing the whole world with 16 pixels. And now the state of art is probably 1,000, 2,000, right? And without considering any complex dynamics of the neurons in the visual cortex, right? But when you do use this very simple approach, and when you look at what people can do with blind, in blind patients, right? So at least they can give the blind people the sense it's bright or it's dark, right? With multiple pixels, they can even, by moving their head, they can even detect edge. They can find out where the door is, right, in front of like a bright background. So that's something useful, right? Although nobody believes that already fully replicates the visual function, right? So.
Thank you.
Can
Steve, you
I
wanna jump
say
in?
something? Yeah. Is my mic working okay?
Yep.
Great. I wanted to, suggest that whole brain emulation might happen the soonest by taking the sort of Ted Berger approach that they did with just CA3 or some small part of the hippocampus for an entire person. Imagine if you could record all of the sensory inputs into every one of their sensory systems, and you could record from every one of their motor neurons, or even just the muscles, from soon after they were born. My guess is, if you had a system that was halfway decent, like AlphaGo, for example, it just looked at a lot of people playing Go and learned from them, and then played itself after that using what it had learned, and developed a very sophisticated ability to behave the way a very good Go player behaves. So
that, as far as I know, didn't include any neuroscience at all, or modeling of ion channels, or circuits, or any of that stuff. You know, it was just input-output functions. So I'm wondering what the other panelists think. Is this a feasible approach to brain emulation, to just put recording on all of the inputs and the outputs of a person and not worry too much about all the levels underneath?
So I guess to follow up on that question, when you think that we can just have this black box and then put in the input and then get the accurate output, it assumes that nothing changes inside, right? So if there's some kind of internal dynamics going on based on some kind of memory from the past and all of these different factors, then given the same input, the output could be different depending on the internal state. So how could you account for something like that in this view?
Yeah, well, you would, if you were following it across a lifetime, you would get a lot of memory effects. You would need to obviously pay attention to what happened in the distant past. You know, that's the whole idea of recording for a very long time, getting a lifetime of input-output functions, some of which the input comes in and the output only comes out years and years later.
Right. So in that case, the input that goes into this black box would not be just the present input at that precise moment, but all the entire history, right?
Yeah.
Yeah. So it's a non-Markov process.
Yeah, well, it's a really long Markov process.
Yeah.
Yeah,
I mean,
sorry. I mean, there's also issue of non-stationarity, right? The system is changing. So now we are talking about input-output mapping that constantly changing, right? So even you have a very long recording, that theoretically won't guarantee you, to get the complete input-output mapping.
Well, I would also be skeptical of that approach, although I've often thought that would be a wonderful experiment, and you could actually cook up ways of you could imagine doing something like that with a symbolic chess playing program being modeled by a neural network program and see what you get. But I think there is a crucial question that we're dancing around, which is what it feels like to be inside, the consciousness question. And I am a big believer that consciousness is a very particular form of self-modeling in the brain, that human beings have evolved to have these brain states that keep track of what it is like to be. And so when I'm often arguing with somebody about well, how do you know if you get somebody's entire connectome, or you get even more information from a scan and then emulate it? And yes, maybe they'll be able to act like the person, they'll act like they're conscious, but there might not be anybody home. I look at them and say, well, we're not talking about just the input-output. We're talking about the internal states as well being replicated. And so, if we believe that there's any insight that we have in our consciousness, where we can say, no, look, I'm really in pain, and that really is connected to the fact that we're experiencing pain, then all of that has to be going through these same levels of spiky neurons. It's a computation like anything else. And so having the emulating it from the level of not just the external behavior, but from the internal representations as well, that I think is a solid argument that if it acts at the end of the day like it is conscious, then it is conscious. Because it would be really hard to believe otherwise. That, and Randall, you were talking about tests, and of course the obvious tests are asking the thing, does it feel right? Does it feel right from the inside? And if they give truthful answers, then you should believe them. Now, if we look at that same thing, but it's the suggestion that Steve just gave, where you've got a massive deep neural network that has essentially learned how to mimic all of the input-output behavior, then even I would be skeptical that maybe what we have here is a philosophical zombie. Because I could believe that there is a point in computation space that acts like a philosophical zombie, but if you look at the internal workings of it, there's nobody home. But I can't believe that that would occur if you actually start from the connectome up, if you will.
Yeah, this is really a goals question again, because I think that the example of the Go player, it makes total sense from the point of view of artificial intelligence, because you're just trying to capture that, right? You don't want the artificial intelligence to necessarily experience it like a Go player. And it also even makes sense for artificial retinas, and it makes sense for artificial hippocampi. Because even if you are replacing the hippocampus with an artificial one, and perhaps it does not capture the entire complexity of the rich experience of having a perfect hippocampus, you still get the same thing that you're getting with the blind patient who's getting an artificial retina. Suddenly, at least they can do something with it, and the rest of their brain is still there. So from that point of view, these tools, they all work building them that way. But I agree with Ken that if your goal is specifically that you want to be able to capture the internal cognitive experience, then you have to care about things that you may not capture from the data that's going in and out, because it's actually encoded in the blueprint of how the brain was developed.
So I think I have some perspective on this consciousness issue. Obviously, it's a subjective phenomenon, so it's very difficult to investigate this scientifically. But I guess there is at least some way of investigating in a scientific manner with n equals 1 or at most n equals 2. So that is, if you can experiment with your own brain. Let's say if you can use neural links surgery device and implant some wires into your brain, then you have some kind of switchboard where you can say, I want to activate this part of my brain, or I want to activate that part of the brain, and just feel what is the change in the subjective experience. Like qualia, when I press this button, suddenly red feels like green, right? And also, this will help you solve all of these issues in validating whether you have successful emulation or not. So you hook up your brain with an emulation device. Let's say you replace your entire left hemisphere with an emulation device, and you still keep it on, right? And you have a switch, and when you flip the switch, then your right hemisphere, the biological right hemisphere, will be interacting with your emulation. And then when you flip the switch again, your right biological hemisphere will be interacting with your left biological hemisphere, and you see whether you feel anything different as you switch between these two modes, right? So something like that can be done at an individual level. So I guess this is very important, because when you think about consciousness in science of consciousness, you're always assuming that it's from a third person perspective. So you're trying to, let's say, look at someone else's connectome, right? And trying to understand and emulate it and simulate it and see whether this is conscious or not. But again, you won't get to any satisfactory conclusion because of the subjective nature. And so I propose that the only way you can go about doing this is experiment with your own brain in some safe manner, right?
Yeah. I'm actually really interested in the, I guess, smaller version of this and the results. Not of the entire hemisphere being temporarily replaced, But just say, for instance, in the experiments that Dong Song has been involved with, to know how patients reported their experience when they were experiencing either the actual hippocampal prosthesis being active or not being activated, or a sham, because of course all of those different options were used in the experimental protocol.
Oh yeah.
So I wonder, Dong Song, how did people report that it felt?
Yeah, they didn't report anything actually. They are not, they didn't get special feelings. So this is very different from some early DBS study, right? Like the one was done, I believe, in Toronto, right? So when they stimulate certain parts of the neocortex, that evokes some kind of sensation of past memory. But what we did with the model-driven stimulation, we use very low intensity, and we stimulate, a smaller number of neurons. So of course we were curious about the patient's feeling, right? And we often always ask that question, but they seldom report there's any, specific feeling associated with those stimulation. Yeah, and by the way, actually, this is a very interesting question. I'm working with another person from University of Washington. We try to do this more formally. So we got a supplementary, grant underneath a study on neuroethics, right? So in future, we want to always include this kind of interview with patients, So what we did previously is just like when the patient take a break and after the experiment, we ask them, right, in a very informal way. But in the future, we want to do this, more systematically. But obviously, there are still a lot of issues, right? The main approach for doing this is basically interview. We ask questions, we get answers. Then there's also issue with about like, how well, how much you can trust those answers, right? So when a patient said like, I feel like a totally different person, right? Is that real, right? Can we have a like more subjective measure of whether he's the same person, right? Instead of just relying on like the personal report, right? So yeah.
I know that you probably all have responses to that, and there are also still some questions from the audience, but I wanted to quickly just throw out there what I think is probably some of the main check boxes that we need to cross off in this this workshop. So it may be a problem that we're preaching to the choir here, and that we didn't invite enough naysayers, or any really. So you see, one of the checkboxes I wanted to go through is, does anyone believe that there is a real case to be made that scale separation is impossible for the purposes of brain emulation? And I'll say the other one as well right away, which is there anyone here who is not satisfied with the level that we're talking at right now? Because we're talking about spikes. We're talking about experiences based on spike patterns. Does anybody here feel that we're not capturing something because we're not going deep enough? So those two questions, the depth and the separability. Yes, Steve.
Yeah, so I would remind some people who, I started out as a biochemist and moved my way sort of up the size scale and also, I suppose, in terms of neural processing scale. I would remind people who aren't too familiar with biochemistry that all of those little molecules that you see beautiful computer rendering graphics of are actually jiggling around and moving extremely fast. They're oscillating in the sort of terahertz range. And ion channels are opening and closing in a stochastic way extremely fast, much faster than we can even measure probably. So I think it's possible. I wouldn't say, I don't really have a definite opinion whether it's likely or not, but it's quite possible that all of that very fast movement, you could just say it's Brownian random motion, is actually a really important part of the computation that the neural system is doing. In which case it's going to be a bitch to emulate all of that stuff. You know, we're talking about a lot more details that we have to worry about. Think about just how difficult it has been to figure out how a protein folds, you know, when it comes out of a ribosome. It's being battered around by all those forces that I was just talking about. It's moving around in this very same way. When we see protein structures, they were made in a crystal, you know, in a very static way. That's the only way we can take a picture of them and figure out what their structure is to crystallize them. When you actually look at them in their liquid state, you know, in the transmembrane state or floating through the cytosol or whatever, they're moving around and changing constantly. So that, getting back to the temporal dynamics, maybe the scales that we have to worry about are actually much, much faster than this firing of action potentials that most computer scientists think, oh, well, neurons are only firing at 100 hertz or so, so you only have to model them at 100 hertz or so. Let's not worry too much about anything faster than that. But this, I would argue that action potentials are not a binary thing. There's a lot of subtlety in every single action potential in terms of its shape and the timing of between action potentials has a lot of analog variation to it. It may all depend on those really, really fine scale, very fast movements of all the important molecules there.
But by definition, I mean, you use Brownian motion as an example. If our model and assumption of what brains actually do is process information, then any effect which is essentially by definition random is not contributing to the sort of aggregation of informational dynamics. It may be having the effect of introducing noise and randomness into a system, which, you know, has its own sort of dynamic effects. You know, the fact that bird flocking is subjected to the sort of random noise of the system imparts these sort of, you know, emergent, you know, the reason the flock, you know, sweeps to the left or sweeps to the right might be some aggregated, you know, sort of bubbling up of the noise, but it's not easily argued that there's information in the noise that is actually having a, whatever we would want to call a meaningful difference. I mean, I'm saying practically by definition, if we're talking about random noise, what possible good can it be if our assumption of what the brain's doing is processing information?
Yeah, my take on it is that random noise, stochasticity, has a clear purpose when it's happening within a set of rules. And that's pretty much what you see in fractals, for instance. But that it can't intrinsically add information to the system. It can only perturb the system, but the system's rules, how the system is set up, is then what determines what might happen, the range of things that might happen.
I think Steve is saying something much more deep than random noise, and you're echoing some stuff that I've heard from other quarters that talk about we shouldn't look to synapses as storing memories because you've got all of this extra biochemistry that could be storing memories inside of a single cell. And I let me throw this out as a counter. I think that the problem is the fan in and the fan out. That a neural system like we see in the brain has the ability to integrate information from tens of thousands of neurons and have that propagate within just a few hundred milliseconds across the entire brain. And so do these molecular interactions have the fan in and fan out necessary to compete at all with the level of computation that we're talking about? As a question to you, Steve, I think.
Okay, I don't think, I think that it's an overwhelming problem to get it all down, to figure out what what are the fan ins, you know, what, which level of information we really need to care about is overwhelming. And I've said before, you just need to focus your question. What are you trying to emulate? If you want to focus on consciousness or something, your job's going to be much harder than if you just want to focus on how does the retina send in information that something's moving in the periphery. So
is that is it? I mean, you're making a hard statement that that consciousness exists at a different level than information flow in the retina, and
yeah,
I think a lot of us here are making the implicit assumption that consciousness is exactly at that same level. It's a different it's different information that's being processed, but it is exactly the same level of information.
yeah, I think level may be not the right word. I would say it's just something that we know much less about. Where does it come from? The stuff that's closer to the periphery, whether it's on the motor side or the sensory side, are things that we just know a lot more about the physiology of.
Let me try to argue. I'm trying. I'm not going to do good. Everything that we know about our consciousness is to some extent us looking back at our previous consciousness using our memory. So to the extent that we know how we have the ability to compare what I'm seeing right now to what was previously stored, that constrains, I think, the computations that are going on in consciousness, and in the sense that if I look back in memory and I saw something completely different than what I'm seeing right now, it's like, oh, this green that I'm seeing right now has nothing to do with the previous green that I remember. Then maybe we would have evidence that that consciousness is at a different style of computation, the microtubules or something, rather than the way that we know memory is encoded, which again is structural changes in neurons that can be accessed with spikes. So is that a way of teasing apart this this question of really we don't know anything about consciousness? Because I think we do know something.
I hope I didn't imply that. Obviously, there are a lot of people that have been working hard on this, and when you narrowly define the type of consciousness that you're talking about, we know specific things about it. You know, if you're talking about visual awareness, that's one kind. If you're talking about attention mechanisms, that's another kind. If you're talking about remembering, recall, you know, I feel like consciousness is really a big giant umbrella that has so many different little adaptations underneath it that you shouldn't really talk about it as a unified thing. It may feel like a unified thing to us, but I don't think it really is. I think that's an illusion. And, you know, we know specific things about certain of those adaptations and those circuits, but there's big concepts that we don't get. So you know, to get at the thing about comparing what's stored with what's coming into the sensory system right now, okay, there's feedback from memory circuits in the cortex that go to the visual processing in V1 while you're perceiving. But what do they do there? You know, what if there's feedback coming from my temporal lobes from when I was a child, some memories that were to do with my childhood experience that are influencing the way I perceive things right now? How does that work? I think we really have no idea how that works. So that's just an example of I feel that our ignorance gets much worse when you talk about things that are further away from the periphery, the motor systems and the sensory systems. It doesn't mean that we don't know anything. It just means that modeling those things, it's an open question right now, how much detail we're going to need to make headway on that.
So do you suggest that we might need a different level of detail for peripheral circuits as we might need for these more higher level, if you will, circuits?
Yeah. So for example, I think I have this intuition that these kind of higher level things that we like to talk about, to do with consciousness, not necessarily just consciousness, but the stuff that's far away from the periphery. We will need, as I mentioned in my interview part, some sort of a whole new vocabulary of dynamical systems to talk about those. That there is a lot to do with attractors that are dynamic, that our mental circuits are traveling along these high dimensional landscapes, and they're going into basins of attraction, and they're going over various different ridges to get to another place. That's to do with coming up with an idea or perceiving somebody and recognizing who they are. Whereas if you're talking about just processing as something moving left or right on my retina, I don't think we're going to need that many high-level dynamical systems vocabulary to describe those things. I think it'll be a lot. It is already. We've made a lot of progress without using that kind of terminology. The people who I'm most inspired by, who seem to really understand this, were Gerald Edelman and Walter Freeman. So, they see. Unfortunately, they're not very good writers. And so to take what their understanding was out of what they've written has been very difficult for me, at least anyway. But I get this intuition when I talked to both of them, when I think Walter Freeman is still alive, I'm not sure, but anyway, I've talked to both of them, and I get this real feeling that they have an intuitive grasp about those dynamical systems way of thinking and what the brain's doing at these very high levels, far from the periphery of the motor systems and the sensory systems.
Oh, by the way, Walter Freeman passed away a few years ago.
Oh, he did.
But I also was very impressed by his theory, especially about the dynamics and the internal dynamics. So his view was very different from the input-output view, and he put a lot of emphasis on what is the internal dynamics and how that actually causes the action, and then that goes back into the sensory system. So everything starts not from the input, but from inside the internal dynamics, and that was the important thing he, I guess, he proposed.
Yeah, but there's one possibility that the more complex dynamics of behavior in the higher level brain region, right? It's just a phenomenon, not the real underlying mechanism, right? But if you think about deep neural network, if you go to the higher level of the deep neural network, you also observe higher and higher level of complexity, dynamics, higher and higher level invariant representation, right? Like the first layer, you see like those Gabor filter, like reception field, and when you go to high level, you can even get some kind of like concept formation, like you see cat, you see human face, right? But yeah, that's obviously true, right? That's also very consistent with what you observe from human. But if you look at the neural network, every layer, they are all the same. And if you look at the input-output function, they are all the same. It's not like, right? It's not like it involves new mechanism or new dynamic representation, right? The input-output function remains the same. You just repeat that building block, right, with this kind of hierarchy. I'm saying this is what I believe, right? But that's a possibility. All the complex dynamics is just a byproduct of this repetition of the simple input-output functions.
Yeah, exactly. So I guess that's a very important point. But the thing is that whether these things that are a byproduct of the underlying mechanism, are these byproducts causal in themselves? So is it just epiphenomenal, to use, I guess, a philosophical language? The epiphenomenal means it's some kind of phenomena, but doesn't have any causal effect. It's just like the smoke from a steam engine, right? The smoke does not drive the steam engine. it's not causal, but it's that indicator that this steam engine is moving. So I guess there's a lot of these issues when you talk about different levels, right? When you have a certain level, and then there are certain emergent properties. So whether emergence is real or not, or causal, is again a huge debate. But we have to think about these things, whether these emergent phenomena are causal in themselves. In that case, how are these different from the level of description that we are modeling?
Yeah, I think one of the things that I noticed when I heard Steve just mention his description of what makes, say, the internal, the deeper systems and how they compute different from, say, movement on the retina and how that's being processed. And then I compare that with what you're saying, Yunsuk, and also what you're saying, Dongsong, is of course they are the same types of circuits. There's the same cells. But there, in my opinion, there is definitely something different going on between, say, the retrieval of a memory from long-term memory, like say it's just the sets of synapses that are they're reactivating a certain pattern, or say the activation of cells as there is light moving across my retina, and then that's automatically processing something, versus what I would call the internal experience of consciousness in the moment. And to me, it feels like a heck of a lot of that has to do with something simple like working memory, which isn't necessarily dependent on special circuitry. It can be how these circuits are operating in nested oscillations or something like that all across the brain. So it's a feature of how neurons work, and that allows you to have that in the moment experience of consciousness. So that's my takeaway typically, but it's just one way of thinking about it.
I mean, I definitely want to second that. I mean, I think that people get off track when they're looking for a correlation between, let's say, a neural firing pattern and an experience. I think that is impossible. That what we actually can get to is a description of how something creates a self model and how it thinks that it is conscious and how it feels like it is conscious. But if you try to nail that down to some kind of this is the experience right now of red, divorced from all the other interactions with working memory and stuff like that, I think that you'll never get to it. And I think that this is where people may come down on this level issue, right? Because if we imagine, let me just throw a question out there. So let's say there was a molecular level terahertz jiggling of molecules that is really doing some incredibly important computation. Can we think of, can we expand that out in any way other than just saying that it's an epiphenomenon? Because if it's an epiphenomenon, it's like, oh, that jiggling of the this particular molecule just is the color red. Then it doesn't interact with memory. It doesn't interact with behavior. It's an epiphenomenon by definition. And I think that we can rule that out on the basis of my inner experience doesn't suggest that there are feelings that I have that I can never express in actions. Okay, and so if we are assuming another level, let's just go down and assume another level, proof by contradiction, if you will. What would that other level look like? And could it be really, how complex would it be to essentially wrap a few differential equations around that and have it interact with the spiking level?
So can I make a stab at an analogy to think about that might help answer that question? I was in the park yesterday, and there's these plants called fireweeds here in Ireland that are growing now at this late time in the summer, about as tall as I am. And at the very top is their seeds that blow in the wind, and the seed is about half a millimeter across, a tiniest little thing. Somewhere in that tiny half a millimeter, with a little piece of fluff connected to it to carry it in the wind, is the design of this amazing, beautiful plant that has these beautiful purple flowers in the summertime. And so perhaps that terahertz oscillation in neural circuits is like that little seed there. You know, it's something with potential to express itself in a much bigger and more beautiful and amazing way, but it's actually extremely compact little piece of something you know, that we're not seeing why that thing is important, but later on it blooms somehow.
Well, with that, if we assume that, sorry.
Yeah,
I'm sorry. If we assume that, does that put a temporal constraint on it? I'm sorry, I may have screwed up the audio here.
Yeah, we hear you.
So I'm suggesting that if there is some little thing that that is occurring, does that mean that whatever computation, let's say, is happening there is only going to be felt by the rest of the system 30 minutes later, for example?
Yeah, I think you can look at how long it takes for that diffusion to happen, and I think you made a very good point earlier that you require action potentials to get information across large pieces of brain tissue, you know, and to get them to react in milliseconds. So you can sort of rule out diffusion and say, okay, let the slowness of diffusion and the time course that we're talking about for things that diffuse are not going to be too important for any phenomenon that you know takes a lot of different brain regions to work.
I was just thinking to link this back to the fundamental question of this workshop, scale separation and how do different scales relate to one another. In order to make a statement about whether these terahertz oscillations have any functional impact, could we define as a criterion for that what impact it has on adjacent levels? So for example, could we emulate very well the spiking behavior of a single cell, of a single neuron, by just using a continuum approximation or somehow making a simplification that does not include these oscillations? Would the spiking behavior be invariant to that? And then, I mean, yeah, so just it's taking spiking here as an example of, let's say, one adjacent level, one level higher in abstraction.
Yeah, so I think I'll go back to our success at modeling using a very non-biological simulation, you know, when we still got some interesting behavior that emulated what we saw in our cultured networks. Probably those kind of influences at the molecular level can be ignored for most of the purposes that we care about. It just depends. The problem is that we don't really know yet. You know, until we have good computers that we can use to model these things in great detail, we won't be able to turn it off and on and do the proper A/B testing. So we'll just have to hope now how we are with our not very powerful modeling systems that we can throw in as much detail as we need to answer the questions that we care about.
Yeah, it's maybe related to this hypothetical case of having a very smart AI study the inputs and outputs of a network, then one of the questions that might come up is, have you studied the network under a sufficiently complete set of input conditions? Maybe you can replicate the spiking behavior of the neuron very precisely, but only for your particular chosen set of input stimuli.
Yeah.
I think there's a connected question that I might as well bring up at this point. In the chat that's sort of going on alongside this conversation, Joe Strout, who of course could also chime in, pointed out that he thinks that compartmental models may be necessary, which, when you start talking about that, then you're talking about, okay, the morphology of the neurons and exactly where spikes appear matters. So where on your dendrite are they coming in? And that seems like another way of looking at this question of which level should we be talking about, because if we care about the effect on the eventual postsynaptic spiking, then you can say, yes, these computations happening somewhere down there, they might matter. And I know that Dong Song has looked into that a bit, because in addition to your hippocampal model, you also deal with a much more complex, large model. Could you maybe point out what you've learned there?
Yeah, so we have been trying to simplify compartment model with what I call nonlinear dynamical multi-input single output model, right? So we try to replicate the spiking behavior by ignoring all the detailed Hutchins-Huxley, keyboard theory based differential equations, right? So, but what we did is a very specific approach. We use like a polynomial, nonlinear function. We use basis function, take advantage of the continuity of the EPSP and the internal variables. So it really depends on what you want to do, right? If you want to replicate the input-output mapping for very limited, spiking behavior for small number of neurons, and also you can tolerate a certain level of jittering or stochastic behavior, then we can call a model successful. But of course, it's not a complete replication of the input-output model, right? Of the input-output behavior of the compartment model. But for a lot of simulation, it's sufficient, right? So, and also, I don't think my approach is the best approach, right? In any sense, or it's an optimal approach. But theoretically, this is definitely something possible, right? Because if you look at compartment model, no matter how complex it is, how many inputs it has, right? How many different differential equations it has, right? They are just differential equations, right? They are already a written input-output mapping, and there's so much redundancy and like degeneracy, right? In this kind of system, there are obvious ways to simplify them. So yeah, I think my answer to this question is that there's definitely ways of simplifying detailed compartment model into a much simpler mathematical form, right? And all those issues, like a lot of phenomena, like which I don't have in my model right now, is I don't have a explicit dendrite, right? And the dendritic structure obviously important, but if you already decided the location of your synaptic input in the specific dendritic tree, but when you look at spiking behavior and the contribution from that input to the overall spiking behavior, it's already fixed, right? Once you decide the location, it's not like it's always moving around, right? Then you have to explicitly model that location all the time. But once it's there, it's fixed, right? So obviously that's information. The information about the specific location can be ignored. It's just the contribution of that specific location need to be embedded in your mathematical representation.
I'm not sure if Stratus, is he still here?
doesn't that ultimately boil down to the same thing, though? If you ignore it, but still manage to incorporate it into your equations, that's a contradiction, right? If it's in the equations, then it's somehow incorporated.
Yeah, but you can either explicitly incorporate it or implicitly incorporate, right? I guess the difference is whether you incorporate the functional contribution of that specific component to the overall input-output mapping, right? If you incorporate it functionally, then by definition, right, when you look at the input-output mapping, you don't see a difference. So, yeah.
So if everyone doesn't mind, I'm going to take a moment to start peppering you with questions that came from, I guess, maybe people who haven't been as deeply involved in constantly thinking about these questions as some of us have. And so that means we can have to step out of our bubble and explain a little bit why we think the way we think. And I'll start with Adam Huzar, who just asked, I was wondering, well, actually, he could say it himself if he wants to, but otherwise, Adam, I'll just go ahead and ask your question for you. I was wondering, what do you all think about simulation of emotional levels that are affected by diverse molecules in the brain? For example, being excited or being sleepy. Do you think we'll be able to capture those effects with, for instance, compartmental models, or a more detailed level is needed, or is a more detailed level needed?
I think we don't even need to get this level of detail of a compartmental model to make great progress on that, because those a lot of that modulatory circuitry is not very complicated or spatially precise. You know, they have axon branches that go all over the place and release neuromodulators like dopamine or norepinephrine over very big regions of the brain. And so they can be modeled, you know, with just a few parameters that say turn up the excitation or turn down the inhibition or whatever. Shimon Marome has done some really good electrophysiology in vitro to show that, you know, the effect of dopamine on a circuit has this kind of like raising the temperature of the system, shakes things up a bit and allows it to settle back down into a new state. So that's something that could easily be modeled without worrying too much about compartments of individual neurons.
Thanks, Steve. I think that probably answered the question. I'm not sure if anyone else needed to jump on that one as well. I believe we have a bunch more. I just haven't been paying attention to the sheet just recently. Let's see. Oh yeah. So Priyanka is asking about nonlinear multiplicative dendritic integration. Yeah, sort of, I mean, the original question was comparing this with artificial neural networks, where typically linear summation is used. But this ties back to the questions about compartmental models and whether you need them or not. Because certainly what Dongsong said about, you know, if you've already included in the effect that your PSP can have on the membrane potential, if you've included in that whatever the effect would be of its distance, say on the dendrite or something like that, that doesn't necessarily take into account something like multiplicative effects or other effects where, say, a spike happening somewhere closer to the soma will interrupt or impact any spikes that are trying to travel there from distal dendrites. So that could be a reason why you might want to build in some more complexity. And I'd be curious to know if, say, the simplification that you've created, Dongsong, whether that takes that into account as well.
Yeah, so my specific approach is instead of using a linear model, right? We have a nonlinear activation function, right? Which is very commonly used technique. So So the specific active function we use is basically polynomial. We use Volterra series, right? So when you look at the polynomial, that's a linear term. The second term is already a multiplicative, right? It's a multiplication between inputs. And then you have a cubic, then you have a quadratic, right? So yeah, that can be included in this kind of model. And also when you look at the more commonly used sigmoidal activation function, right? That can also be easily implemented in that. When you look at sigmoidal, that's a sublinear phase, right? That's a linear phase. Then that's a that's a superlinear phase. That's a linear phase. Then there's a sublinear phase. And the multiplication can be sort of approximated by that superlinear phase, right? And also, as we know, right, it's mathematically proved that if you have a sigmoidal activation function, and if you have more than 3 layers, you can approximate, or actually you can fully replicate any arbitrary input-output mapping, right? So, I think there's no reason that cannot be done mathematically, right? And also, I want to repeat what I just said about the location. So if the location of that spike, that synapse and the other synapse, they are activity dependent, then it will be really complicated. You have to explicitly include that location into your model. But once the location is decided, that interaction is just whatever the interaction is, right? There must be a simplified description of that effect is, even it's complicated, right? So in that sense, you don't really have to include that location information or the nonlinear interaction in that very small segment of the dendritic tree in your model. What you care about is what that location does to the output signal, right? If it's multiplicative, then you have multiplication in your model. If it's additive, you have linear term in your model. So you don't have to explain why that happened. What you need is to describe or capture what already happened.
Thank you. And we do have one question from Dan Breeden and from the YouTube chat, and he asks, would the integrated information theory of Tononi allow mind uploading in principle? And I'm not sure who would be, I'm not sure who to ask to answer this one.
I volunteer, Keith.
Oh, please, thank you.
Yeah, well, I mean, any number probably able to answer that question. I think Tononi has said that he's actually basically okay with uploading so long as it's done his way, which is basically no simulations allowed. I don't think he said that he thinks that there's some sort of, prohibition in the universe against uploading. It's just got to be this ITT thing sort of going along. That's what I thought. If anyone feels like I've miscaptured him, jump in.
I've heard the same thing from Christoph Koch, so I believe that is their position on it. Although you can't really get a more clear idea of what level that they would require for their analog simulation. I have a different overriding question, if I could. So there's two ways of taking this level question. There's the question of what is the level that we're going to have to make sure that we capture in our initial kind of simulation of the brain to make sure we get everything, okay? And this is kind of what Dong Song was saying. So it's initially you might have to know where each synapse was on the dendritic branch, for example. But then there's a possibly much more interesting question of what is the minimum level that you will eventually get to once you start dragging out the redundancies and stuff. What is the minimal level that will capture human cognition and consciousness? So I think it's best to explicitly state both of those and kind of relate questions to those. Does that make sense, Randall?
Yeah, that makes total sense. Sorry, Michael. Yeah,
Okay.
you can continue asking questions. I'm not going to. I think it's great for me to step back and let others be moderator for a while, just because then other types of questions get a turn. But I do agree with you completely, Ken. Of course, there is a huge difference between what is the simplest, most efficient system that I could possibly run this equation on, or something, or what I can transform this equation into that will be more efficient, right? And this is that completely makes sense. And at the same time, you may need to look to a much more detailed, more complex data set to get there, because the original biological system that you're getting it from was implementing it differently, right? And so you need to make that translation happen, and translations can be costly. And that's actually where my other big concern is about, the model fitting problem in machine learning, because that's basically about translations between equations from one set of data to a different set of data, namely your parameters.
And just to pitch in on the question about the relevance of IIT on mind uploading, I guess IIT, as far as I understand, it can tell you whether something is conscious, whether it's you or your simulation. Okay, so it can tell you whether successful emulation is conscious or not, but it may not be able to tell you whether you and your consciousness and the emulated consciousness, these two are identical. So I guess that was a topic of the previous workshop about identity. So I guess I'll stop there.
Yeah, along those lines, there's IIT is saying that there is, I guess, a computation. It's subtle, but there are people that don't think that that computation captures at all, and I guess they're not represented at this workshop.
Can I?
I don't think so.
Oh,
yeah,
sorry. I just one thing about the question of compartments and localization of synapses and multiplicative versus additive interactions and so on. So maybe the level of detail of compartments is an interesting one, in the sense that it's quite a continuum. So typically how we first get to a compartmental model is that it's based on a reconstruction that some poor PhD student did in a lab somewhere, spending hours bent over the microscope and adjusting the focal depth and clicking some points along the dendritic tree. So really it already begins with a kind of discretization, and could also use EM scanning and it would have a finer resolution, but there would be some discretization to begin with. And then we could gradually make that compartmental model simpler and simpler by having two adjacent compartments, by merging them together, and then merging together larger and larger sections. And probably for a while as we do this, the functional input-output behavior of the neuron will be relatively unchanged, again with the caveat we're looking at a certain subset of input patterns. But we could we have a knob that we can tune to bring the complexity down almost to a neuron with maybe even two compartments, one compartment representing the soma and one the apical dendrite, for example. So it might be, and so I thought this was interesting to contrast it with other levels of detail discussed at the workshop here, which seem more of a binary thing, like do we need the terahertz oscillations, do we need the microtubule, and so on. Here we have more of a continuum, it seems.
Okay, so follow up on that point. As you reduce and reduce and reduce and merge, and assume it to be a continuum, do you have to worry about things like bifurcation? Let's say in the logistic equation, as you change the parameter value by minute amounts, at a certain point, suddenly you get period doubling and then chaos, and then back to some different periodic behavior. So if it is truly a continuum, and the assumption there would be that the transition between these whatever scales would be smooth, right? But what if it's not like that? If there's some kind of fractal property to it.
Yeah, at some point you've simplified it too much and it, it goes out of whack.
Yeah,
absolutely. Yeah.
Yeah, I think that the current approach to simplify compartment model is still to use compartment model, right? Hutchins-based keyboard theory based equations to represent those reduced compartments. The other possibility is you change that, because mathematically, multiple compartments cannot be fully replicated by a simple compartment. Some information is losing, but you may change you may make that single compartment more powerful, so it's possible to capture multiple compartments behavior, not necessarily using Hutchins-Huxley based equation anymore.
Yeah, I don't know if Michael or Bill are currently searching for other questions from the audience. That gives me one more chance to ask for the second time, because we didn't quite exactly get everyone's sort of buy-in or buy-out from this one. The separability question. So we've talked a lot about the level, but is there anyone, at least in this audience, who would like to make a case or at least propose a situation where we would not feel that scale separability is possible at all if you want to preserve the experience of cognitive function? So basically, this project is a failure from the start. Or maybe we all believe it is possible, that's why we're here, and we're, computational neuroscientists.
Well, I'm not going to disagree, but that there's not level separability. But I would say that I often get this point where somebody says, even if you had an exact copy, that exact copy would start to change immediately, and therefore it wouldn't be the same. And so any of these questions have to come with a caveat that says good enough is good enough. If there is no good enough is good enough, then yes, there's no level of separability that can satisfy that.
Yeah, I fully agree. I think the cost function of the matrix, right? The matrix for evaluating what's successful, that's really the key, right? If you care about every single detail, right, you want to fully replicate every behavior of a complex system, then I guess by definition you have to, right, use all the details. But it really depends on what you want to do. And my belief, right, I cannot prove it. It's I think the answer is definitely yes. And if we use human cognition, human behavior, or even human consciousness as that cost function, I even want to say, I believe that can all be achieved at the spiking neuron level. So
maybe
you can't insist that the two systems receive the same sensory input. So, right, I mean, for that reason alone, you can't say that they are, even if they were conceptually deterministically identical, they would diverge behavior because they're receiving some variation in input system. Like the whole notion is just off the table.
So maybe a better way of asking this, Randall, is if you were dying of X and you could be uploaded at the level of spiking neurons, would you go for that today? And so I'll throw
So
out a
if you're
yes
asking,
for myself.
yeah, you're asking me directly, and I will of course also answer yes. It's a, you know, we don't even have to go that far to ask the question. With death, it's a, you know, that's a pretty big one. But even, you know, if somebody can't form new episodic memory, would you be happy with a somewhat functional hippocampal prosthesis as opposed to a perfect one, or none? Yeah, sure. You know, if you can't get a perfect one, then take what you can get. And I think it's the same sort of thing when it comes to even the entire brain. Let's just say, to me anyway, like even for the entire brain, even a, you know, unless it turned out to be a really bad situation where you made such a crappy model that it meant that I was constantly in pain or something, which I doubt very much because I could see how you could immediately solve that problem in an emulation. But yeah, barring those sorts of things, absolutely yes. I'll take a spiking one.
And there are other levels that, you know, I would immediately dismiss as completely crazy, like, you know, 1 millimeter MRI scan of the brain, structural scan or something. I would say not a chance. Don't even waste my time with it, right? So I think that might be a different way to figure out people's commitment to to this level separability problem.
Any other takers of Ken's question? Ken's version of my question? No one wants to go on the record? You are taking notes, right, Ken? And you're going to make it into a manifesto, and then you're going to put it out there, and it's going to be like, these are all the neuroscientists who say, and then blah blah blah. Right.
Yeah, I think his point was that, you know, I agree with that. You have to define what it is that you're happy with, and once you define that, as long as it's not everything, you don't need to. You can do separation of levels. You can say, all right, I'm just going to do anything that's synapses and above, action potentials and above, you know, probably not cubic brain millimeters and above, but you can get a lot done with action potentials. We know that because we've, you know, there's been a lot of actual electrophysiology, not even talking about the models, just the electrophysiology and what people can predict in terms of what animals are about to do or what they're perceiving from the action potentials.
Well, I mean, let me. It seems in this, it seems obvious, you know, again, that we're kind of all talking about the same level. But when I go back to my, you know, you know, I'm sorry, I shouldn't bring this into this discussion, but I think it may be relevant. You know, we have a brain preservation technique that can preserve the connectome, that can preserve the ultrastructures of synapses, that can preserve ion channel densities and receptor densities because it's glutaraldehyde based. We have it preserves messenger RNA expression levels, et cetera, and yet when I ask neuroscientists, is that sufficient for the possibility of future uploading? Is that sufficient for the possibility of future uploading? Which is, I submit, is the exact same question that we're addressing today. I get, we don't know enough. How can you even start to talk about stuff like this because we don't know enough about the brain? And so, so I can either assume that, number one, that there really is a debate within the neuroscience community, that there might be some really deeper levels that we're not even addressing, that even every ion channel would not hit, or people are really uncomfortable about saying that computation has anything to do with consciousness. Or that people are just, when hit with the ramifications of this is not something that's going to happen 200 years from now, but this is something that we can start the first step on today. and their mind is blown, and then they have to search for a reason to argue against it.
Well, Ken, here's one objection from a neuroscientist. You're missing the temporal dynamics when you fix something and study it under the EM. The current trajectory of what's going on in the brain might be important.
But we know that that isn't important, though, because we know that you can do profound hypothermic circulatory arrest procedures on human beings where it shuts down essentially all pattern neural activity for 30 minutes. And we do that in surgery. So there is no possibility that you've got some dynamics, or am I missing something?
That's a good argument. I would like to see somebody revived from having been actually frozen, to the point where everything was in a solid state like the way it is in the EM, and then revived and see if they still remember what happened 5 minutes before that.
Well, okay, so we have to, again, it's about sort of deciding what we're all sort of in agreement is the critical sort of function that you can or cannot lose. To be, quote, frozen or sort of not frozen, but kind of cryonically, I've lost the word for freezing without shattering all the cell walls. But Ken's example is that in rapid frigid drowning and intentional, sort of hypothermic, thank you, intentional medical procedures, usually performed not for a brain emergency, but actually for a cardiac emergency. You just want to get everything cold so you have enough time to sort of put the heart back together, quote, quote. In these examples, yes, it's not freezing. I don't know who decided that, sort of the physical freezing point of water is somehow apropos to the discussion. It's just some arbitrary number picked from physics. But what we do know is that in these examples, by certain criteria, the brain is not doing certain things. And one thing it's definitely not doing is firing any action potentials. Okay, billions and billions of neurons, and there isn't a single action potential site for hours. And this is just a fact. So if we're going to say that a certain, sort of cutoff in functionality has been satisfied and then been recovered from, then you have to sort of fold that into your argument, either for or against whether or not this works. We do know medically it works. In fact, we know psychologically it works. These people have revived and they feel themselves to be a recovered patient. Their doctors and families regard them as recovered patients. No one goes around sort of rewriting the metaphysical philosophy of how our society is going to treat people and just sort of assigning them new identities. Like, video is frozen. Did I lose my internet? Shoot. Anyway, I hope my point came through
still
before
there.
my internet dropped down.
Yeah, you came through. It was a little halting, but you came through. But yeah, no, you're right. I mean, I think obviously we've been kind of too fast to judge all of those hypothermic procedures successful, and just to be cautious, we should take away people's identities and say that they're no longer married to their spouse and remove their house and whatever after they've gone through this procedure, because then they're someone else, right?
So I have a question to Ken, which is so related to Steve's question about the temporal dynamics, keeping the temporal information. My question is, from your EM data, can you reconstruct some temporal parameters, such as integration, membrane integration time, and all of these dynamical information? I guess delay is somewhat you can estimate based on the diameter and whether the axon is, let's say, myelinated or not. But what about these intrinsic temporal parameters? Can you reconstruct that?
Well, I think the question is, can we construct it today versus can it be constructed in principle?
Oh, yeah, in principle, let's say.
And so you would have to ask, well, what is being lost? And something that has been discussed, I think, a couple of times today, is that the extracellular space is typically lost in these in the EM preservation procedures. So to the extent that that really carries information that is crucial and cannot be is not held redundantly, then maybe there are certainly some things that could in principle not be reconstructed. But at the same time, I can point out a whole bunch of papers that that do, for example, compare directly the correlation between synapse size, dendritic spine size and stuff, and the electrophysiology. They'll take two neurons, as a recent paper takes two neurons, does electrophysiology between them, finds the synapse that is between them, and then does EM. Did that for a whole bunch of them, and plotted the size in EM versus the electrophysiological response. So I think there are arguments that that say you can get a heck of a lot if you're imaging everything at 10 by 10 by 10 nanometers.
Do you need to worry about the genetic makeup? For example, Henry Markram did a lot of work on this, right? So the neurons that look morphologically exactly the same would have different physiological properties dependent on the different kind of genes that they express.
Absolutely, but they're going to express, they're going to have different genes turned on, which means that they're going to have different messenger RNA expressions, which means that they're going to have, more appropriately, they're going to have different ion channel distributions. And so I'm not saying that you would not, I think that you probably would be able from EM to get neuron type information, and that would be good enough. But at the same time, our preservation techniques today can certainly preserve ion channels. Can't image them today. I mean, we can't image with good EM and simultaneously all the different ion channel information, but our preservation techniques are certainly good enough to preserve these things.
I noticed that Dong Song turned on his microphone a while ago, so I was wondering if we kept interrupting before he was able to say something, or if that was just a coincidence.
No.
Okay.
Yeah, but yeah, actually, I wanted to say something about, so if you ask most of the neuroscientists, right, we do believe all the memory, all the internal states are stored in the structure and the synaptic weight, right? Which in principle can be reconstructed by just looking at the like EM data, right? So, but there could be, one possibility that maybe, but I'm not saying this is a reality, right? If some information, some memory, or some of the identity is stored in some kind of persistent electrical activity, then you probably can lose that information, like some little oscillation that right, that will just die away if so.
Right, you have amnesia if you get a concussion, right?
Yeah.
Right.
So then we're right back at, we keep on losing our identity every time these sorts of things happen. Yeah.
Mm-hmm. And the one related topic that I remember Ichkovich, right? He often makes give the example showing why timing is important, right? Because if you simulate a very large scale network by changing slightly a position of a single spike, because of the chaotic behavior, eventually the activity become totally different, right? With or without that change in the whole brain, right? But my understanding to this phenomenon is that this exactly explains why this chaotic behavior is not important, right? Because right, you can easily change the position of a spike just by probably just knocking out your head, right? But you are still yourself, right? So there must be some high-level invariant property that with more resistant to this kind of eternal perturbation.
Right.
So yeah.
Yeah, that was actually another major part of what I hope this conversation would get into, but we never really got into a strong discussion about complex systems and chaos theory and all of that. But yeah, that's I think what we're looking at here is a system that wants to be understood. My memory systems want to be able to talk to my visual system, want to be able to talk to other systems. They don't want to lose the ability to communicate just because there are some stochastic components in the way that synapses communicate. And similarly, every time I see a tiger, I want to be able to run away from it and not sometimes think it's my grandmother. So there are some real evolutionary pressures for the system, even if it's based on a long string of analog nonlinear components, to make itself not so chaotic or not unpredictable. You know, more like a chaotic system in the sense that a system may have a huge range of possible behaviors, but that that range is still a range that you will permit, one that is the one that you want the system to be able to operate in. And then you've got things like recurrent inhibition and oscillatory rhythms that make sure that patterns happen at certain times and that they are phase offset from patterns in another area at a certain way, and that you have populations of neurons instead of single neurons, so you don't have to depend on the spike time of an individual neuron, and you've got bursts instead of just spikes sometimes because you want to make more robust communication, and so forth and so forth. This is why when I did the little intro video at the beginning, I was kind of explicitly comparing, which by the way, I'm sorry for the production quality, we did it last night, explicitly comparing the very huge billions of nonlinear analog transistor systems that we use every day called computers with this other billions of nonlinear analog systems, analog component system, the brain, where the computer is making itself completely predictable, totally deterministic because of the rules imposed upon that nonlinear analog system, and that the brain has to do something rather similar if it should fulfill its function of keeping living organisms alive.
So Randall, I just to loop back around on the big question of separability. Is it better to, if the goal is whole brain emulation, you can look at that as essentially what it would take to make a de novo brain, or what it would take to copy a brain. And if you look at it from what would it take to make a de novo brain, then you are essentially asking the question of what's the minimum set of things you would have to deal with. And that is an unstructured problem right now. It's highly unstructured, right? If you and at the end of the day, you're going to get something that is not satisfying to anybody. It's going to be like, well, some level of computation. The alternative way of looking at this, how to copy, what would it take to copy a brain, is saying what level of resolution and molecular identity would you have to do to scan a brain to make sure that you emulate it. And I think that is a well-structured question that we can address today.
Yeah, I totally agree. And that latter one is indeed actually the topic today, because this is, this.
Oh my God, I'm
really the main
sorry.
topic,
I
right?
should have read the. I should have read the syllabus. I'm sorry.
No, you're very well stated, Ken. Thanks.
At this point, we can bring in a question from the YouTube live stream. Glenn Clayton asks, do we need to understand how the brain produces consciousness to emulate that consciousness? It would seem that understanding how a system works is not necessarily required to emulate the system. I pasted it in the chat.
I think we have to have a theory. One of the big problems here, and I'm not sure many people are really willing to admit this, is that it actually be, to some extent, unsolvable. it's possible that those of us who are sort of physicalists, functionalists, materialists, sort of believers in all of this, we may not be able to prove that we're right. Or, it may not be possible to prove that you're creating a philosophical zombie if you satisfy all your physical and neurological requirements. And you just have to say that your personal preference for sort of, how you model the world is that, replicating certain behaviors and sort of phenomena that you can measure and determine from outside. You're just saying, well, we think that this can't be faked. So our model is that this is a conscious system. And someone else who's just planted their flag in a camp where you cannot be conscious if you're not biological, or you cannot be conscious if you're a simulation, a physical net underlying Coke goes on about. You can't be conscious if you don't have microtubules going on, whatever. I don't know if you can just sort of prove anybody into changing their mind about any of this. We may just have to sort of separate ourselves into cults and kind of, maybe that's where the truth is, these very cults and these.
Okay, well, I want to weigh in on that by
Something just happened to the audio.
saying again something that I've tried to harp on before, that I feel consciousness comes in different degrees, and you can decide how much of it you want to emulate. I think the greatest thing about IIT is that it allows you to quantify just how conscious anything is. So my claim that there are many different levels of it, you can prove it numerically using IIT. You can actually measure quantitatively how conscious is this person while they're sleeping, how conscious is this animal, how conscious is this computer program, by probing it using that theorem. So I think if you want to talk about can we even emulate it, you have to first say, how much consciousness do you want? And I don't think, because I believe that the brain operates under physical principles, I don't think there's any level that we couldn't eventually emulate. And I suspect, based on the experiment that Ken describes of freezing people's brains and reawakening them and finding that they're pretty much the way they were before, that the temporal dynamics isn't something that we need to worry too much about when we're recording all the details that we need to put into our emulation. So given that, I think we may have all the tools we need to emulate. Say it again.
It's not freezing, certainly. It's lowering temperature down to 30 degrees Celsius during a profound hypothermic and circulatory arrest surgery. Just want to be clear.
Okay, well, some animals do vitrification. I think aren't there some that they can actually they produce their own antifreeze compounds and they become vitrified and revive themselves after the thaw.
But how do we know that's not a different frog every time?
Yeah, it might could well be a different frog. It's hard to ask them.
And my point is that the question is baseless. It's, first of all, it's a philosophical question, not a scientific or medical question. Second of all, it's unfalsifiable. Like, it doesn't mean anything to ask the question. To conclude that it's a different frog or the same frog, if your whole understanding of science and nature is empirical measurements of physical phenomena, to conclude that metaphysical identity undergoes a transformation regardless of what you can measure experimentally is by definition an unfalsifiable question. And I think we just have to I think we just got to push that whole mess off the table. And we here in this room can choose to do so. I don't know how we convince sort of society at large to just accept that. I think that this is because the question you're asking is very popular, and and I really don't believe that we're going to make any ground on it, because it's not subject to the march of advancing science. It just doesn't work that way. I don't think we're going to get anywhere. I think our philosophy might grow. You know, in the next 100 years, society's philosophical approach to this may undergo a transformation, but we're not going to solve the problem. I honestly don't think this question works that way, is my opinion.
Keith, so would then the alternative be to, in making a decision on how to use the success criteria to determine a successful whole brain emulation, if we have to accept the fact that that we might not have a complete understanding of, let's just say, consciousness, for example, as a success criteria, do we just have to then just use what we are able to experimentally sample, like for example, like just.
Well, I don't believe that we're going to be overly convinced of a success by neural tracings. I think that Randall has described this well before. I think what you're going to have to do is interview the damn thing. And if you convince yourself that you're talking to, you know, essentially, if you basically, it's a Turing test. It's the argument that you can't fake the Turing test. If the system convinces you via interview that it is the revived patient, then it's the same experience you have when you interview a revived hypothermic patient. You just have to decide that because they present a convincing symbol of the prior person, you're just buying. And that's, I don't think you can do any better than that, basically.
Can I argue against that? Sorry. So I think what Yunsuk said earlier, I mean, he gave some examples of, you know, when you get brain interfaces good enough to start switching in and out. And we're, you know, if you look at Neuralink and you project that out 50, 100 years, I think the general person on the street is going to have the ability to switch in and out a piece of silicon and feel what it is like to have that piece of silicon either in them or not in them, and maybe even, yeah. So I would suggest that there are ways to enlighten society regarding the philosophical zombie question.
I didn't speak up when you said that earlier, but I think Daniel Dennett would disagree strongly that you can subjectively feel differences in your own qualia. The whole point of the sort of dancing qualia experiments is that you can flip back and forth, and you don't know you're having a different qualitative experience. It's not that red and blue, red and green, whatever the canonical, it's not that they feel different. It's that they don't feel different, but right. The whole point is that there's some sort of fundamental difference in the qualia.
But that's
isn't there some major reason?
I'm sorry. I thought. Sorry, but the audio cut out for a second, and I thought you were done.
Yeah, I think that there are certain experiments which can change your visual qualia. Let's say it's all related to the movement. So whenever you move to the right, let's say suppose you present some blue tinted thing. Right? And if you do that consistently, whenever you move, I forgot about the details about this experiment, but there are certain cases where your motion gets related to a certain qualitative experience of color. And because you move your eye in a certain way after this kind of training period, now your qualia would slightly change and you can detect that. So I'll find the reference and share it later. But anyway, so there are certain experiments like that I'm aware of.
Yeah, I think that is another example of how important embodiment is and the environment, that those things are what lead to all those qualia. Qualia are not just something that's in your head. It's to do with all your interactions with the world outside and all the experiences you've had with it.
But even with the change in qualia, would that change the answer of the first person perspective? Because the point to be able, it seems like the point to be able to have that interface connection is so that the first person can say, yeah, it worked. But even if the qualia did change, like from a third person's perspective, they might be able to notice that it changed, but would that change the validity of the first person's perspective, which was the point of the interface?
That's a very good question. I don't have any clear answer to that at this moment.
It's sort of, let me pose an interesting question in sort of speculating about radical alterations to your senses. Let's say that we had the necessary sort of neural medical control, prosthetic control, to take a red-green colorblind person and temporarily correct it for them. Okay, so you flip the switch, and they distinguish between red and green, and they have two different qualitative experiences looking at, you know, Christmas pictures mostly. And then you turn the switch back off again. And they're looking out at the world, and the red and the green look the same. And to those of us who aren't colorblind, it doesn't mean anything to say that they look red or they look green. It doesn't mean anything. It's just, it's whatever the color red-green is to a color blind person. Okay. Now, this same person, reflecting on, remembering the experience of the switch being flipped. I pose it as an open question. I don't know the answer. What the hell for them are experiencing red and green differently if they cannot experience it now that the switch has been turned off? I'm not sure that that's a valid question, which is why I'm not sure, you know, philosophical experiments, so we flip switches on our qualitative experience. I'm not convinced that this makes sense.
Well, you'd have to specify if you're flipping what you're changing is in the retina or if it's in the color area of the brain, like what you get in achromatopsia. Because you know, if you destroy a part of the brain, people not only lose the ability to see color, but they lose the ability to imagine the differences, I believe, if I'm getting that right. So, I mean, you know, I would argue that this is a much more complex system that is being oversimplified by the idea that you could just flip the switch.
Yeah, I don't know if we're finished with this question yet, but if we
Never.
are, let me know, because we have another one coming in from the audience. Anyone still want to follow up on this question? Okay, so we're not going to manage to completely solve the question of how to test for same qualia or different qualia. Okay. So I'm not 100% sure how to interpret the next question. Dan Breeden was posing something where he asks, you know, are the properties of networks the only things we need to emulate? And when I probed a little further to find out exactly what he meant by that in terms of what are the other things besides properties of networks, what he really meant was the networks of neural systems are the basis of first-person perspective, not individual components. So the networks as opposed to the individual components. What would be your responses to that?
Networks of transistors add numbers in computers. The individual transistors don't.
Yeah, that.
Well, my bias is to think that networks are really important, and we should be spending a lot more time thinking about neural systems at the networks level, and building more tools to study them at the networks level. So yeah, that's maybe just because that's what we focused on in my lab, but I think there's a lot of network dynamics that we haven't even touched yet. That there are subtleties there that we're just beginning to even see, let alone understand.
And I think to a certain extent, these types of questions, if they had been posed 20 years ago, would be they'd have a different feel, because with the deep neural network revolution that we're seeing today, we can see exactly what networks are able to do and how little individual components play in those processes. It really is the case that when AlphaGo plays, it doesn't matter what any single node is doing in that system. It's all about the network behavior.
Yeah, I think obviously this is the same answer that most of us or all of us are going to give in a sense, one way or another. But I get the feeling that his question was more about when listening to this discussion, it seems like we're talking about, should we be recording neurons? Should we be recording the size of dendrites and where synapses are arriving? Should we be recording, other messengers and whatnot? But what about the system level where, things are really happening? So why are we focused on what we're going to record at that level down there? Maybe it's obvious to us and not to the listeners. Well, I guess I'll give my answer, which is, yeah, if you're going to study what the networks are doing, or you're going to recreate what the networks are doing, yeah, how do you do that? If you just want to describe the network at a very high level perspective, coming up with an equation that describes what the entire network is doing, yeah, either you're going to do something very abstract where you end up with, oh, this is a brain oscillation, or you're going to try to do something detailed, and then you can try to do it detailed in what we might have just started calling the de novo brain method, where you can make up how you want to describe what your network is doing in terms of its parts, in terms of parameters of some form. Or you can do so by describing how that network is doing what it does in a complex way or in an emergent way, in the form of parameters that you might be able to reconstruct or measure from your measurements. So parameters that you can actually identify or that you can describe based on what you can identify in the biology. And that's why we always end up going back, of course, to neurons and synapses and all of that, even though what we really care about is the network level. But then we also, we describe problems such as, if you have a gazillion things that you've just measured, how do you turn that into a system that does something useful? And in order to make it do something useful, you need to know how you can tell whether your system, the way you're tuning those parameters in your system is leading to something that is what you might call successful outcome, something that behaves like a brain or behaves like a hippocampus. And for that, you do need to have some blueprint that you're comparing with, some typical type of behavior. So you do need to know something about how the system behaves at that higher level. Because only if you know how those systems behave at that higher level, then you can tell, how to correct for errors as you're modeling your system, or whether you're even going in the right direction. And so, yeah, I guess we all agree with Dan Breeden that you need more than just the neurons, you need the networks too. Okay, Joe, yes, I think we're all kind of feeling it. I'm getting that sense from the way, we're getting to our questions more slowly, and perhaps we're sort of wondering, well, wasn't this supposed to wrap up at around 12:15, 12:25 or so? And you're right. This isn't the only time we're going to have conversations like this. This is part of a series, so maybe this is a great time to wrap it up. And we've done a lot of talking, so I'm not going to make it much longer. But I do want to thank everyone involved. First, I'll start by thanking the panelists who are here and those of you who have agreed to interviews, whether your interview has already been shown or is still going to show up later, because there are a few we've already gotten into or at least prepared for that haven't been part of this workshop. And so Steve, thank you very much. And Ken, thank you very much. Dong Song, Yoon Suk Cho, Keith, Charles, and I'll mention a few others who haven't yet had their interview shown, like Naveed Farahani and a few others. It's wonderful that you care so much about this problem, that you're willing to dedicate time on a weekend and time before that to just try to walk us through your thinking so we can compare notes and maybe make some progress. And as I mentioned, this is going to be an ongoing series for a while as we are mapping out the new roadmap. And so there will be more. We're going to analyze what came out of the session today, and we're going to pinpoint a few things that might be the right topic to dig deeper on. I also really want to thank Michael Ulrich and Bill Yuen, because you guys just put in some fantastic effort preparing this session and preparing for interviews and things, and many others. So many people on our team have contributed in one way or another, or continue to do so throughout the year. So thank you very, very much, everyone. And with that, if anyone wants to stick around and just chit chat, please do. But I'm done.
I guess at this point, we can address some closing remarks and some reminders. Once again, we are a 501c3 nonprofit operating with the sole mission of advancing research in whole brain emulation. We appreciate the many contributions that have allowed us to organize events such as these. If you would like to become a monthly donor or to make a one-time donation, you can visit carboncopies.org/donate and donate by PayPal, credit card, bank transfer, or cryptocurrency. You can also contribute by volunteering. We are always looking for those who have a technical background to help with our technical review, and anyone who has experience in the nonprofit world, such as in recruiting, event coordination, fundraising, marketing, and so on. You can go to carboncopies.org/openings to apply. You can also sign up to our newsletter or follow us on social media at our Carbon Copies Facebook group, facebook.com/groups/carboncopies, at the Carbon Copies Reddit page, reddit.com/r/carboncopies, at the Carbon Copies Twitter account. Our Twitter handle is @carboncopies.org. And as always, all audience members are invited to take our survey. The survey link is in the video description. Thank you all very much, and hopefully we'll see you all again at our next event.
Thank you very much.
All right. So